hello, namaskara, vanakkam. Welcome to Shalini Simplifies. In this video, we're going to learn about how plants that produce flowers will make little babies of their own. The name of the chapter is Sexual Reproduction in Flowering Plants. This is the first chapter in your 12th grade biology syllabus, and a lot of students do not like this chapter. They find it very boring. They can't relate because it's not about humans. But you know what? This chapter is so entertaining. It has more drama than most Bollywood films. Similar to how in most movies you have a hero and then there's a love interest and they fall in love and a lot of drama unfolds. Even in this chapter, you can expect a lot of entertainment. Okay, if understood properly, this is very similar to a love story. So, in today's class, we will try and fall in love with this chapter. Even if you don't fall in love with this chapter, you must force yourself to, because this is important. You can expect as many as 3 to 4 questions to appear from this chapter alone in your NEET exam. So, you can't afford to not like it. So, let's hope to make this chapter easy and memorable. Let's get started. Now, let's begin by trying to decode the name of the chapter. It's called Sexual Reproduction in Flowering Plants. Obviously, for most of you, it is self-explanatory. But let me just quickly tell you what it's about. Clearly, it is about reproduction, which is why it is included under the unit reproduction. What is reproduction? Reproduction is just another biological process similar to circulation, respiration, excretion, digestion that you can see occurring in living organisms. What does the term reproduction refer to? If you can split it, re means again and production means something is getting produced. It's a general English word. So reproduction basically is a process by which something is getting produced again and again and again. So in the context of biology, we basically talk about how living organisms get produced generation after generation, and the process by which that happens is called reproduction. Okay, so broadly, when you classify reproduction, you have 2 different types. There's asexual reproduction and sexual reproduction. Earlier in your syllabus, there used to be a chapter called Reproduction in Organisms, where you learnt about the difference between asexual, sexual and all of that. But after the syllabus got rationalized, we no longer learn about it. So here, I'll tell you briefly what asexual reproduction is with respect to flowering plants, plants that produce flowers or angiosperms. Whenever you get plants similar to the parent by using parts of the plant other than the flower. Like sometimes, let's say you go to your neighbor's house and you find a plant in your house that's producing beautiful yellow color flowers and you want that in your house. Let's say it's a rose plant. So what do? You don't wait for the flower to produce seeds and you don't go get seeds and then sow them, right? So instead you take a stem cutting, come and put it inside a pot, and that will give rise to a new plant. In that case, you did not wait for seeds to form. You did not use seeds for getting your new plant. Reproduction happened by using a vegetative part of the plant. Similarly, you can use roots, you can use leaf buds in order to get new plants. For as long as you're getting newer plants by using parts of the plant other than the flower or the products of a flower or like a seed or something, then you can call it asexual reproduction. There are numerous ways in which that can be done, but that's not the focus of this chapter. In this chapter, we're going to focus on sexual reproduction specifically. So what is sexual reproduction? How is it any different from asexual reproduction? See, in asexual reproduction, when you're using a plant part like a stem, a root or leaf buds or whatever, the offspring that you obtain, the newer plants that you obtain will be identical to the parent, exact clones, like all of the gene combinations are going to be the same, exact same. No change, absolutely. But in sexual reproduction, that is not the case. The reason there is so much variation around us, one of the major causes for that is sexual reproduction. So in sexual reproduction, meiotic cell divisions will take place. As a result of meiotic cell divisions, newer gene combinations arise in the offspring, and the baby plants don't look exactly like the parent plants. Not look, of course they might look similar, but they will not have the same gene combinations like the parent plants. Okay, so here meiosis happens, gamete formation happens, gamete fusion happens. So there's a lot more complexity involved. And in this chapter, we're going to focus on sexual reproduction in flowering plants. Okay, so we're going to learn only about how sexual reproduction happens. It's very similar to how in humans a male gamete sperm fuses with the female gamete egg and a zygote is formed. All of those processes will happen here as well. Okay, so that's about how sexual reproduction takes place briefly. And here they have specified that it is in flowering plants. What are these flowering plants? Flowering plants are nothing but our pretty angiosperms. Angiosperms commonly are called flowering plants. You know how in the chapter Plant Kingdom in 11th grade, you've learnt about 5 different divisions of plants. We've discussed algae, bryophytes, pteridophytes, gymnosperms. All of these have been discussed in absolute detail, their life cycle, everything we've learnt in the chapter Plant Kingdom. But angiosperms was just briefly spoken about. I always tell you in 11th grade that after Plant Kingdom chapter, there are 3 chapters specifically dedicated to angiosperms of flowering plants, 2 of which you would have finished in your 11th grade syllabus, which is morphology of flowering plants and anatomy of flowering plants. And in your 12th grade syllabus, we are talking about how sexual reproduction happens in flowering plants. Remember, not all plants produce flowers. Gymnosperms like the Christmas tree and a you don't see flowers blooming on them. Pteridophytes like ferns and bryophytes, little mosses, they don't produce flowers. The flower producing plants are called angiosperms. This is the most advanced group of plants you can find on our planet. Also the most dominant, more number of plants are angiosperms itself. So we will talk about how sexual reproduction happens in angiosperms. That's going to be the focus of this chapter. Now talking about flowering plants, obviously you have to start talking about flowers, right? Flowers are super important in our daily lives. There are so many floral fragrances we swear by, like you can't step out of the house without your fragrances, like your perfumes or whatever, most of which are the fragrances obtained from flowers. And then to decorate our hair and do hairstyles, we make use of flowers. In worshipping God, we make use of flowers. In expressing love, we make use of flowers. When somebody passes away, then also flowers are used. So basically flowers are so versatile in their utility. They are used pretty much everywhere very commonly. So they have become an important part of our lives, and also you can't imagine nature without flowers, right? It'll be like very, very boring and very, very just greenish, different shades of green. Because of flowers, there's so much color and vibrance in nature. So flowers are very, very important for us. But in this story, we're going to learn about why are flowers important to plants. Obviously, we have come up with numerous ways to use flowers, but it's not, the plants don't produce flowers so that we can keep it in flower vase at home, right? There is a reason why any plant produces any part. There is a reason why leaves are formed. There is a reason why plants have roots. There is also a reason why plants produce flowers, and it's not for us to use it. So the plant has some benefit because of flowers, which is why they produce them. And what is the benefit of flowers to plants is that it is the organ that helps in sexual reproduction. Okay, so if you look at humans, when we talk about the reproductive parts or the male reproductive parts and the female reproductive parts and all of that, you always talk about the testes and ovaries as primary sex organs, right? Of course, the penis is involved in reproduction. Of course, the uterus is what bears the baby. But why do you call the testes and the ovaries as the primary sex organs? That's because for sexual reproduction to happen, the gametes are very, very essential. The male gamete sperm and the female gamete egg are essential. It's only when these 2 cells fuse will you get the first cell of the baby, right? So whichever part of the body, whichever organ gives rise to, these gametes is referred to as the primary sex organ. Likewise, in plants, it is within the flowers that the reproductive structures like the spores and the gametes are formed. Therefore, the flower is called the sexual reproductive organ. In this chapter, we are talking about sexual reproduction in flowering plants, and the reason flowers are produced in plants is because that is the place, that is the organ in which sexual reproduction takes place. More about it we will learn as we move on. Since this is the first chapter in the unit reproduction, there is a little bit of unit introduction at the beginning of every unit. Questions can arise from that part too, so we can't afford to skip it. Let's just quickly go through what is given in the unit introduction and then we will move on with the chapter. Alright, since this is the first chapter, obviously they're talking a little bit about biology. So they're saying that biology in essence is the story of life on earth. Of course it is, it's a beautiful story. What is biology? It's a branch of science that deals with living organisms. So they are trying to, write it like poetry. So it's given that it's the story of life on earth, how life evolved on earth, how living organisms are, how they reproduce, and a whole lot of other things you will learn about in biology. So what they're trying to say here is, they're trying to explain the significance of reproduction in living organisms. See, we have to understand it with respect to two contexts. Firstly, for an individual organism, is it essential? Absolutely. And for a population, what is the role that reproduction plays? You see that a lot of living organisms do not reproduce. Some of them choose not to have babies or not to have offspring, while in some organisms, they cannot. Okay, for example, if you consider a mule, which is a hybrid animal formed when a donkey and a horse mates, that is sterile by nature. It cannot have children or it cannot reproduce. But if you look at many human couples, some of them choose not to have children. That does not mean, reproduction is, that basically means that reproduction is not essential for an organism to survive. If I have to survive, it's not necessary that I have to reproduce. Even if I don't reproduce, I can live and then I can, at one point, everybody dies and people die. That's all. But if a whole population of humans stops reproducing, then what would happen? That would be very bad for the human species, right? For the human population to continue to exist, reproduction must happen. For one individual organism, it's not absolutely necessary for survival, but for the survival of a species, reproduction is essential. If reproduction does not happen, a species cannot survive. Why, you ask? It's common sense, right? All living organisms they come with a limited lifespan, right? It's not like we are immortal beings. It's not like we can live for 2000 years or millions of years. The reason we have been existing since probably thousands of years is because of the process reproduction. If we have a limited lifespan, everybody that is born is going to sooner or later die. Once all living organisms die and they have not reproduced, obviously the species is going to go extinct, right? So that should not happen. Ultimately, all living organisms on our planet, starting from bacteria to plants, animals, fungi, every one of us have one biological purpose. It's not to become a doctor, it's not to become like an awesome teacher or anything. It's to make sure that even after we are gone, our species continue to exist. Over the course of evolution, in order to, enhance our reproductive fitness, many organisms have adapted in many different ways. More about that we will learn in other chapters. What I'm trying to tell you here is reproduction is essential for the continuation of a species, but not essential for the survival of an individual organism. Okay, so because why it's essential for the survival of the species is because when individual organisms die, when we die, without fail, of course, the species will still continue to live through millions of years unless it is threatened by any other forces. So reproduction becomes a vital process. In this case, if not for reproduction, the species cannot survive for a long period of time. Reproduction can happen either by asexual or sexual means. I've told you about this. I've told you the advantage of sexual reproduction too. Sexual reproduction makes sure that there is a variation in the population. Not everybody is like a clone. When there is a lot of variation, for example, if you consider a human population, we are all humans. We have a pair of eyes, a pair of hands and a pair of legs, and all of that is similar, but still we have a lot of differences with respect to the color of our eyes, how tall we'll grow, what is the skin color, what is the hair texture, and all of that, right? So that is called variation. Within a population, despite having similarities, the differences that we have is what is called variation, and that arises because of sexual reproduction. Why is variation important? Why can't everybody look the same? Why can't everybody be the same? It's not good for the species as a whole, because whenever there are more variations in a population, if at all there is any environmental disturbances or anything, it's like some individuals will have some gene combinations that can resist that, and they can adapt better, and that way the species can survive. Okay, so that's the whole idea. Always, if there is variation in a population, that is advantageous for that particular species. All right. So in this chapter, we're going to learn about how sexual reproduction happens in flowering plants. We learn about how in this unit, and the next chapter is going to be about how reproduction happens in humans, which is a beautiful chapter. And then following that, you have a chapter called reproductive health, in which we will learn about contraception, we will learn about assisted reproductive technologies, which is also very, very interesting. So that's all this unit has to offer. So now we will start with the first chapter, which is sexual reproduction in flowering plants. Proper, like unit introduction is over. Now we'll start with the chapter. Okay. So here we're going to learn about sexual reproduction under three different stages. Like you already know, in most organisms, animals and plants included, one of the hallmark signs of sexual reproduction is the fusion of the male and the female gamete, right? That happens here as well. Do you remember what that process is called? The sperm fusing with the egg cell. What is that process called? When a male gamete and a female gamete fuses with each other, we call it fertilization. Since we're talking about sexual reproduction here, fertilization is a very, very important event. And it is also special in angiosperms because something happens during fertilization, which is unlike anything you've seen in algae, bryophytes, pteridophytes or gymnosperms. So it is a unique process exclusively seen in angiosperms. We call it double fertilization. So during fertilization, the male gamete has to fuse with the female gamete. And as a result, the zygote forms, right? But. We are talking about angiosperms in which the dominant phase is the sporophytic phase, right? The sporophyte itself will not give rise to the gametes. For fertilization to happen, gametes have to be present. They are absolutely essential. But the sporophyte does not directly form the gamete. The sporophyte will form the spores develop into gametophytes, and the gametophytes then give rise to the gametes, right? So, a whole lot of processes are involved for the formation of gametes. Before fertilization takes place, spore formation has to happen, gametophyte formation has to happen, gamete formation has to happen. So, many structures and events have to occur. We'll first learn about those pre-reproductive structures and events. Pre means before. Whatever has to happen before fertilization, we will first understand that. Then we will learn about how exactly this type of double fertilization is special and unique. And then after fertilization, whatever changes happen, that we will learn under post-fertilization structures and events. Post means after, right? So, after fertilization takes place, the zygote develops into the embryo, ovule transforms into the seed, ovary wall transforms into the fruit, and all of that happens. So, that we will learn under post-fertilization structures and events. Now, you know how plants, after they germinate, like when you sow the seeds inside the soil, a baby plant arises. Initially, it will show some amount of vegetative growth, right? First, the root system and the shoot system forms. Initially, there are tiny leaves. Eventually, the plant grows taller and the leaves expand and they become wider in surface area, and all of that happens. Only much later will the flowers form. It's not like as soon as a seed germinates, it will already have flowers. The vegetative parts will start forming immediately after germination, but the flowers will get produced only much later. And if you compare that with humans, it's the same case even in humans. As soon as a baby is born, it will not be able to make more babies. It will not have the ability to reproduce, right? Only after you reach a certain age, like maybe mid to late teen, is when you hit puberty and you gain the ability to have babies. So, what is the reason for that? Why is it that flowers are not produced immediately after a seed germinates? Why is it that leaves are produced but flowers aren't? There are two reasons. Firstly, producing flowers is kind of energetically expensive for the plant, okay? So, initially when the plant is too small, when it has very few leaves, it will not be able to afford that. It will not have that much amount of reserve food to, afford or spend in order to produce a flower. Number one, first reason. The second reason is that if a flower is produced, that means that is an indication that soon there is going to be a baby plant, okay? So after the flower gets produced, fertilization will happen and seed formation will happen. For flowering plants, seeds are like their babies. So if a flower is produced, that means the seed is going to form very soon, and that seed, if it germinates, that's like the baby of the plant. Now in any organism, if you look at, I told you that all of our biological purpose is that our species has to continue even after we are gone. You know how parents are so protective of their children, they want to make sure that everything goes best for their particular child, right? Plants might be brainless, but they're not stupid. They're super clever. They also understand this. They will make sure to produce flowers only after knowing that the conditions around wherever they are, present is favorable. Okay? See, if a plant produces a flower when the conditions around are not favorable, let's say it's too cold, or let's say there's not sufficient water present in the soil, or there could be the light intensity could not be optimum for the plant. If in a condition that is not favorable for the parent plant itself, if it ends up producing a flower by spending a lot of energy and a seed even forms, in such conditions where the temperature could be too high or too low, or the water is less, or there is not sufficient sunlight, do you think the seed will be able to survive such harsh conditions? It won't. Okay, so if there is too much snow or frosting, then the seed will not be able to germinate. Even if it germinates, it will not be able to survive. So plants have a way of determining whether the conditions around are safe or not, because they're spending a lot of energy in producing a flower, in pollination and all of that, right? If that energy goes waste when the conditions are not favorable, it's a loss for the plant. Energy going waste is one thing. Secondly, even if they spend so much energy and produce a flower and end up having a seed, the seed will not be able to survive. It's like the baby of the plant is dead now. Who would want their children to be dead? No organism, right? So the plants, in a very clever way, monitor the environmental conditions, the duration of light, the duration of day length, the temperature and all of that. Only once they are sure of the environmental conditions being favorable for the seed's growth, germination and all of that, only then will they decide to produce flowers. So how will flowers be able to see the duration? It's not like they have a skin that can sense. It's not like they have eyes that can see. How do these stupid plants, monitor all of that? That could be a question. Plants aren't stupid, like I always say. They have a way in which they can determine the day length and also the length of the dark period during night. So only when the conditions are favorable, then they will produce a hormone called florigen. All of this detection mostly is done by the leaves. So leaves have certain chemicals and pigments for the same. So once they're sure that it is a good season to have children, they will send a signal from the leaf in the form of a hormone to the tip of the shoot. Now, what is the tip of a shoot? If this is like a plant, this tip of the plant, stem tip, is called the shoot tip. To the shoot tip, chemicals that are produced by the leaf will be transported. What happens at the shoot tip now? Normally, what happens at the shoot tip? At the shoot tip, normally you can find, like if this is the shoot tip, if you take a small portion at the tip and look at it under the microscope, you will see tiny projections rising from its sides laterally. These are called leaf primordia. Okay. Primordia. Eventually, these will expand and give rise to the leaves. But right now, you do not want leaves anymore. Now the plant has decided that it's going to produce flowers. Instead of leaves, flowers has to form there. So what happens is, instead of producing leaf primordia laterally, the shoot tip will start producing what is called floral primordia. Okay, so firstly, the plant has decided to flower. Many hormones have been transported from the leaves to the tip of the shoot. In flowering, a lot of hormones like gibberellins are involved. Apart from that, also there's a hormone called florigen that will initiate flowering at the shoot tip. Okay, so once this hormone or these hormones reach the tip of the shoot, there will be some structural changes that take place. Structure of the shoot tip will change. How does it change? Instead of producing what are called leaf primordia, it will start producing what is called floral primordium or primordia. Primordium is singular, primordia is plural. So, surrounding the shoot tip, you can see many such floral primordia forming. So, these floral primordia will eventually give rise to the inflorescence. This axis will become the axis of the inflorescence. To this axis, many floral primordia are, kind of attached. Each of these floral primordia will eventually form a floral bud. Initially, it used to form leaves, but right now it's going to form floral buds. And these floral buds will bloom to give rise to our flowers. and within the flowers there will be gynoecium, androecium, the reproductive parts and all of that. Okay, so this is what happens at the tip of the shoot when a plant decides to flower. Even before you see that a flower is formed on the plant would have made the decision based on the environmental conditions. Okay, so only when everything is safe for its future baby will it produce flowers. Now, yeah, so that's what is given here. Now, flowers are modified condensed shoots, we say. What does that mean? What is a shoot? Shoot is the stem part. You know, this is the stem. On the stem, you have specific regions called nodes. These are called nodes. It is at the nodes that leaves are born. In between two regions, in between two nodes, whatever regions of the stem is present, they are called internodes. About this you've learnt in the last, the previous year in a chapter called Morphology of Flowering Plants, right? Yeah. So this is how a normal shoot is. But what is a flower? How is it any different? It's given here that the flower is a modified shoot. First of all, what is a modification? You've learnt about modifications of roots, stem and leaves in 11th grade, same chapter again. Whenever a plant part or a plant organ performs a function other than its primary function, you call it a modification. If a leaf, apart from performing photosynthesis, if it does something else like helping in climbing, then you call it a modification. If a stem, instead of just bearing the appendages, performs the function of photosynthesis, as in the case of Opuntia, you call it a modification. If the root, apart from helping in absorption, is helping in storage of food, you call it modification. Whenever an organ does something other than its actual primary function, you say that that part is modified. Here they are saying that flower is a modified shoot. That means this shoot, apart from, producing leaves that perform photosynthesis, is going to do something else, right? So over here, it said that it is modified, which means that here, instead of producing leaves at the nodes, it's producing parts of the flower. At the lowermost node, you can see that the sepals are forming. The next node will have petals. Further ahead, you will have stamens forming. And higher up, you can see the formation of the carpel. So, what was supposed to be leaves are instead forming parts of the flower. So, calyx is also a modified leaf. Petal is also a modified leaf. Stamens are also modified leaves. Actually, they are modified leaves that bear sporangia. So, these are like microsporophylls. The pistil is like the megasporophyll. All of them are leaves that are modified. But this doesn't look anything like a flower, right? So, they get condensed. What does condensed means? What does condensed mean? It means that the internodal length will decrease. Okay, so in between these nodes, whatever internodal regions are present here, I've shown it in an elongated way here. That length reduces to such an extent it will seem like all of these parts are arising from the same point. So if you look at it, it's going to look like this. You have the calyx or the sepals, then you have the petals, you have the stamen and the pistil, all seeming to arise from the same point. But in fact, they are not arising from the same point. They are all present at different nodes because the internodal segment or the internodal region is condensed so much, it will seem like they are appearing from the same point. Okay. So once the shoot gets modified and condensed, what we get here is our pretty flower. Now, a flower has many different parts. What we are looking at here is the structure of a typical angiospermic flower. You have many modifications. Some flowers have stalks, some of them don't have stalks, some of them have sepals, some of them have sepals and petals. Some of them have just one whorl called tepals. So variation definitely you will see in flowers. Some of them are bisexual, some of them are unisexual. But here what you're looking at is a typical flower. All of the parts that are found here are found in most flowers. Okay, so exceptions of course will exist. So this flower has a stalk, and the stalk of this flower is called the pedicel. At the tip of the pedicel, you can find a broad expanded region, which I'm, highlighting here. This is called the thalamus. So on top of the thalamus, if this is like, you're looking at it, you're looking at a longitudinal section of the flower. If a flower is like this, then we're cutting it like this and looking at it here. But if you look at it from above, okay, you can see that the four parts of a flower, the sepals, the petals, the stamens and carpels, all of these are arranged in the form of concentric circles. I'll show you how. Let me draw it slightly smaller. You have it's arranged in the form of concentric circles like this. Okay, and these concentric circles are called whorls. In the outermost whorl, you have the calyx. Inside that, you have the corolla, which has petals. Inside that, you will find the stamen. And inside that, you will find the pistil, which will have its ovules and stuff. Okay, let me just draw it like this. So this is how it looks from up above. Now, the sepals are the individual units of a flower. Each leaf-like structure is called a sepal, and collectively you call it the calyx. Petals, each individual unit is called a petal. Collectively, the entire whorl is called corolla. Stamens are the individual units of andrecium, which is the collective term. Carpels are the individual units of the gynoecium. Over here, sepals and petals do not have direct involvement in sexual reproduction. They are there, of course, for as long as they are there, it's beneficial for the plant. But even if sepals and petals are absent, reproduction can still take place. But carpels and stamens, or andrecium and gynoecium, these are non-negotiable. The carpels or the gynoecium is the female reproductive part, andrecium is the male reproductive part. It is within the andrecium and gynoecium that the respective spores are produced. As a result, they are absolutely essential for sexual reproduction. And these sepals, I'll tell you what its function is. Of course, you would know it from last year. But sepals, when the flower has not yet bloomed, when it is still like the bud condition, as we saw here, it protects whatever is present inside. What is the function of petals? Petals are usually showy and brightly colored, large. Their function is to attract the pollinators. And about andrecium and gynoecium, I already told you. Remember, andro relates to males. So, andrecium, gynecologist, gyne relates to female. So, it's called gynoecium. Now, since this chapter is about sexual reproduction, we are going to mostly focus on stamens or andrecium and gynoecium. No details about variation in petals and sepals that we've done in the chapter morphology, right? Now, let's move on and talk about alternation of generation before we start talking about the stamens. Alternation of generation is something that you've already learned in 11th grade in the chapter Plant Kingdom. I'll quickly revise that. If you understand that, then this chapter is going to be like a cakewalk for you. So, you know that in angiosperms, the dominant phase is the sporophytic phase. In all plants, you have a sporophytic phase and a gametophytic phase. The sporophytic phase is going to be the diploid phase and the gametophytic phase is going to be the haploid phase. So what happens? How do the sporophyte and gametophyte alternate with each other? The sporophyte produces the spores, spores develop into gametophytes, gametophytes produce gametes, gametes fuse to form the zygote, which eventually gives rise to the sporophyte. Now when it comes to angiosperms, sporophytes produce two different types of spores. Okay. It produces male spores that are called microspores and it produces female spores that are called megaspores. Both of these are single cells, okay? So microspore is haploid, megaspores are also haploid. Microspores and megaspores are both single cells. The single cells themselves will not give rise to the gametophyte. It will not give rise to the gametes. It will give rise to what is called the gametophyte. So microspores give rise to the male gametophyte. Megaspores give rise to the female gametophyte. Gametophytes are also haploid. Now the male gametophyte gives rise to the male gamete, female gametophyte gives rise to the female gamete. Now the male gamete fuses with the female gamete to form what is called the zygote, which is diploid. This diploid zygote will eventually form the sporophyte. So this is what happens in angiosperms. We are now looking at a sporophytic plant. The sporophytic plant has produced flowers. Within the flower, within the male reproductive part, which is the andrecium, microspores will form. Male microspores will form. Spores will develop into the male gametophyte, which will give rise to the male gamete. Within the gynoecium, the sporophyte will form the megaspores. Megaspores will give rise to the female gametophyte. Within the female gametophyte, female gamete will form. Eventually, these both will fuse to give rise to the zygote, which is diploid. Whenever spore formation is happening, I want you to understand that meiotic cell division will take place. Look here, the sporophyte is diploid, but the gametophytes are haploid. Whenever you want to obtain haploid structures from a diploid structure, reductional division or meiosis will take place. So over here during spore formation, meiosis takes place. But look at gamete formation. The gametes are haploid. They are forming from the gametophyte, which is also haploid. Since haploid structure is giving rise to a haploid structure, equational division or mitosis will take place here. Generally, when we talk about what kind of cell division takes place during gamete formation, even while we are differentiating mitosis and meiosis, we usually say that mitosis happens in somatic cells, meiosis happens in, germ cells. Whenever gamete formation happens, meiosis happens. But I want you to remember this very clearly. Whenever gametes are forming from the gametophyte, mitosis will take place. Whenever spores are forming from the sporophyte, here is where meiosis takes place in flowering plants. Alright, so what we're going to do right now is, in the pre-fertilization structure and events, we'll first learn about how is the structure of the stamen, how are the spores produced inside the stamen, the microspores, how do the microspores develop into the male gametophyte, how do the male gametophytes give rise to the male gamete. First we learn about the story of this male hero. Later we learn about the story of the heroine. Then we will talk about how they both come together, form the zygote and all of that. Okay, so now we'll start with the structure of stamen. Now the stamen typically has two parts. This long slender stalk that you see is called the filament, and at the tip you have a knob-like structure that is called the anther. So anther and filament are the two parts of the stamen. If you look at the filament on one end will be attached to the thalamus. Right? What is the thalamus? I just told you that it is on the thalamus that all parts of the flower arise from. See over here, this is the thalamus, this is where the stamen will arise from, right? So, in most cases, the stamen will arise from the thalamus, but in some species like in brinjal, chili and tomatoes, you will notice that the filament is fused with the leaves. I'm sorry, not the leaves, the petals. So, it could arise from the thalamus or the petals as in the case of brinjal family, or it can even arise from the tepals as in the case of lilies. In lilies, you will notice that there is only one non-essential whorl which we call the perianth. It has tepals. To the tepals, the filament is attached. Okay. So, this end of the filament closest to the thalamus, this end is called the proximal end. The other end of this blue filament is called the distal end. Now, what is the function of this filament? What is the function of the anther? We'll understand. Now, at the distal end of the filament, you have this knob-like structure which is called the anther, and the anther is the fertile part of the stamen. What does that mean? It means that I told you that within the stamen, microspores will get produced. Now, the stamen has two parts. Are the microspores going to get produced in the filament, or is the anther responsible for that? It is within the anther that the microspores will get produced, which is why we call the anther the fertile part of the stamen. If the anther was the fertile part, what is the point in having a long filament? Why should it need a stalk? Why can't just the anther be present attached to the thalamus? There is a reason. If the anther is directly attached to the thalamus, if the anther is right here, then when pollen grains are formed inside, when the pollen grains have to be released, they will be released right within the flower and cross-pollination will not be achieved successfully. Because the filament is there, it is long, and it exposes the anther. When the pollen grains are released, the chances for cross-pollination increases. Okay, so that's the reason why filaments exist. And if you look at the length of the stamen and the number of stamens, it varies from species to species. In some species, like in hibiscus, you have numerous stamens. If you look at pea plants, you have 10 stamens per flower. Its number varies, its length also varies, its structure, how is the filament attached to the anther, all of that varies from species to species. Okay. So now that the microspores are going to get produced within the anther, we will learn about the structure of the anther. The anther, as you can see here, has two lobes. This is one lobe, this is the other lobe. So there are two lobes present. Since the anther has two lobes, each anther, per anther there are two lobes, we say that the anther is bilobed. Next, within each lobe of the anther, what you're looking at here is an enlarged version of the anther that is transversely cut. Okay, I'll show you with an example. Now, you can think of this as the anther. This is one lobe, this is one lobe. These two lobes are connected to each other by means of a tissue called connective. Okay, this is one lobe and this is the other lobe. Two lobes are there. They are connected by connective. Now, within each lobe of the anther, you can find two chambers. Okay, so look at this anther lobe. You can see two chambers. Within this anther lobe too, you can find two chambers. Because there are two chambers per lobe, these chambers are called theca. You say that the anther is dithecous. Di meaning two, here again bi means two. Actually, in many reference books, because there are two lobes itself, you call it diathecus, but according to NCERT, since there are two chambers, it is called diathecus. Now, I told you that it is within this anther that spores are forming. So, this is a structure within which spores are forming. There is a technical term to refer to a structure within which spores form. You've learnt about them in other groups of plants, you've learnt about them in fungi. So, what is a structure within which spores form called? Can you think about it? It is called the sporangium. Now, inside each of these lobes, pollen grains will be formed, microspores will be formed. So, each one of them is equivalent to a sporangium. So, within each lobe, you can see that there are two sporangia existing. Totally in one anther, you have 4 sporangia existing. These sporangia give rise to microspores. So, we call them microsporangia. Okay. So, per anther, there are 4 microsporangia. Since there are 4 microsporangia, you call it tetrasporangiate. Tetra means 4. Anther has 2 lobes that are connected to each other by means of a connective. Within each lobe, there are 2 theca. Each of these chambers within the anther will produce microspores. So, you can call them microsporangia. Since there are 4 such chambers, you have 4 microsporangia. That makes this anther tetrasporangiate. Okay. Now, I want you to notice one more thing. If you look at this anther lobe, it's not smooth here. See, it's not smooth here, right? There is a depression or a groove of sorts. It's like this. Look here, there is a depression here. It is not smooth. Now, this is called the longitudinal groove. It's a groove. It's present as long as the length of the anther. That is why it's called the longitudinal groove. Now, this is important. See, once the anther produces pollen grains and the pollen grains mature, eventually the pollen grains will have to leave the anther. So, the wall of the anther will have to break open and release the pollen grains. It is at this line that the anther will break open and release the pollen grains. That is why this is called the line of dehiscence. Dehiscence is the breaking open of anther to release the pollen grains. Since it happens at this line, you call this the line of dehiscence. So, this is the structure of our anther. Now, we have to learn about the structure of the microsporangia. This structure of each microsporangium is what we must understand. So, here you are looking at an anther, a transverse section of an anther. This is how an anther looks. If you cut it, like make an extremely thin slice here and look at it from above, here's what it's going to look like. It has like a butterfly sort of shape. This is one lobe, this is the other lobe. At the center, you have the connective connecting both the lobes. So, we're going to focus on the structure of one microsporangium. Whatever is the structure here, the same thing will apply to the other 3 microsporangia as well. It's very easy and simple to understand. Okay, so one microsporangia is zoomed in and enlarged and shown to you here. Now we must understand one thing. Inside these microsporangia, right inside, like wherever I am outlining these regions, inside these regions, microspores are forming. Okay, so eventually microspores and pollen grains will be formed here. That is why we are calling this whole structure microsporangium, right? So, these are the microspores. Microspores are very important for the plant because it is through the microspores that we get the male gametophyte and the male gamete. So, during the formation of microspores, there should be sufficient protection for the microspores to develop. nourishment is also required. So the other layers of the anther surrounding the region where microspores are produced, this is the wall of the anther. See, I showed you here. This is the wall of the anther. Inside is where the pollen grains will get produced. Now, when I showed you this, it seems like there is empty space here. But in reality, the space inside the anther, the space inside the chambers is not empty. It's not khali here. Within this also there is tissue present. That tissue itself will give rise to the microspores in future. Now, for microspores to form, nutrients are required, nourishment is required, and since they are very precious cells for the plant, support is also required. Both support and nourishment is taken care of by the wall. The wall here in my model anther is very thin and simple, but the actual anther's wall is pretty thick. This wall, it's made up of 4 different types of layers. Okay, so we learn about the different layers that you can find in the wall of an anther. We'll start from the outside. The outermost layer of the anther's wall is called the epidermis. It's not something new. Every part of the plant, root, stem, leaves, everywhere the outermost layer is called the epidermis. Okay, I'm outlining the epidermis in pink color for your reference. I'm doing such a shabby job. Okay, so this is usually a single layer. Right inside the epidermis, you will find the second layer, which is called endothecium. Epi means above, endo means inside. Outermost layer above, that is the epidermis. Endothecium is the layer that is present right inside it. So this is the second layer from outside. This also is usually a single layer. Inside the endothecium, you will find what is called the middle layers. Whatever I'm outlining in green color, that is the middle layers. That is third layer from outside, and these middle layers can be 1 to 3 layers in thickness. And we have the innermost layer, which is, which color have we not used? Yellow. Okay. So this innermost layer is called the tapetum. Tapetum also is generally a single layer. In tapetum, you may find more than one nucleus present. How do you think one single cell can have more than one nuclei? That's because during cell division, first nuclear division happens and following that you will have cytoplasmic division taking place, right? Karyokinesis happens first, following that cytokinesis takes place. In tapetum, karyokinesis happens first. Immediately after that, cytokinesis does not take place. So one cell ends up having more than one nuclei. Now, if you look at the functions of these different wall layers, similar to how in our houses we have walls, right? Walls' ultimate function is to protect. It could be the wall of your house, wall of your school, wall of an office. Anywhere a wall is meant to protect whatever is present inside from the external surroundings. It could be wind, water, animals, whatever. Even over here, the wall has protection as its function. The outermost 3 layers will protect the anther when it is young. When it is young, it needs protection, so it protects it. When the anther matures, what do I mean when I say the anther matures? When the anther matures, pollen grains or microspores are already formed inside. Once pollen grains and microspores are already formed inside, if it's continuing to protect it, if it's being like a rigid wall and protecting what is lying inside, pollen grains can never leave, right? For pollen grains to leave the anther and get outside, the pollen bowl has to break open. and that process is called dehiscence, right? So when the anther is young, it requires protection. Once the pollen grains are mature, the pollen grains have to leave the anther, for which the anther wall has to break open. So the same wall layers that were offering protection to the anther when it was young will break open and release the pollen grains as the anther matures. Okay, so when the anther matures, it will help in the process of dehiscence. Now, what is the function of tapetum? See now, right inside the tapetum, whatever cells are present, these cells inside the tapetum, this is called microspore mother cells or sporogenous tissue. Now, what does sporogenous mean? Sporo relates to microspore in this case. Genus relates to generating or producing. So, it is through these sporogenous tissue or the microspore mother cells that the microspores will form. For the formation of microspores, nourishment is required, and that nourishment is provided by none other than the tapetum. Whatever substances that it will supply to the sporogenous tissue for nourishment, it will release into the sporogenous tissue. So these cells will absorb that, utilize that, and use that to produce microspores. Since this tapetal cell layer is in close proximity to the sporogenous tissue, it is ideal for these cells to support the development of the microspores, right? If the epidermal cells have to offer nourishment to the developing spores, it would become difficult because they are spatially separated. They are far away from each other. The tapetal cell layer and the sporogenous tissue are neighbors. They are very close to each other. So, it is ideal for the tapetum to offer or provide nourishment for the developing spores. Now, these microspore mother cells in short are called MMCs. Since they are mothers of microspores, they are called microspore mother cells. Obviously, they are going to give rise to the microspores, right? So, this is the structure of our microsporangium. Epidermis, endothecium, middle layers and tapetum, all of these are various layers present in the wall of the anther, similar to how the walls in our houses have many layers performing different, functions. Like center, there is a brick wall that provides the framework, and on top of that you have a layer of cement, and on top of that you have a primer, on top of that you have paint. Different layers have different functions. Even in the anther's wall, there are different layers that perform different functions at different times. Okay, so this is the structure of the microsporangium. Our next job is to understand how exactly these cells inside the microsporangium, the sporogenous tissue, the microspore mother cells, produce microspores. And that process is called microsporogenesis. Microsporo clearly relates to microspores, and genesis means generation or production. So it's no big deal to define microsporogenesis. It is a process by which microspores are formed. Where does it take place? It takes place inside the anther. Now, this microsporogenesis happens within the sporogenous tissue, right? So this is our sporogenous tissue. Within the sporogenous tissue, you have numerous cells, right? There are many cells present inside the sporogenous tissue. These are called microspore mother cells. Since these are directly involved in spore formation, that is all I am showing, but the other wall layers, of course, everything will be present inside the anther. Okay, so we'll take out just one cell from here. Whatever happens to that cell, in all of the cells, you will see the same fate. Okay, so let's take out one cell from here. This is the sporogenous tissue. I've taken out one cell from there. This cell is called, let's call this the microspore mother cell. The microspore mother cell is diploid Okay, it is part of the sporophytic plant body, so it is diploid. Now, this is supposed to produce spores. During spore production, what kind of division happens? During spore production, meiotic division will happen. From a diploid cell, you are going to get haploid spores. So, here it is within the microspore mother cells that meiotic division will take place. After meiotic division takes place, 4 microspores are formed. You can see only 3 here. Imagine another one to be behind this. So you have 4 haploid cells formed. Since they are all present together, you call it a microspore tetrad. Tetra means 4. Since there are 4 cells that are present together, you call it microspore tetrad. I'll tell you why these spores are kind of attached to each other and are not separating. The reason is because before microspore formation happens, you know, these porogenous tissue, whatever cells are there, they are neighboring cells. Between neighboring cells in plants, you obviously see cytoplasmic connections, right? So if you think about it like 2 rooms, let's say this is one room, this is one room. In between these 2 rooms, there is a little bit of, let's say there is a door or something through which people from this room can go here, people from this room can come here. So even in cells, it's like that. When they are neighboring cells, substances can move into and out of neighboring cells by means of cytoplasmic connections called plasmodesmata. Before microsporogenesis begins, what happens is each of these cells will form a wall on the inside. They will form a wall on the inside such that the connection is cut off. Now this cell and this cell are not connected to each other. This wall is called the callose wall. It is made up of a substance called callose. So all of these cells on the inside will form thick walls and get rounded off like this by a wall called callose wall. And after this callose wall has formed, microsporogenesis will take place. So initially all of these microspores, all 4 of them, are within a common wall called the callose wall. Callose is a substance which the wall is made up of. Eventually, an enzyme called callase will act on it and this wall will get disintegrated, it will break down and the 4 microspores will get separated. 4 microspores are haploid. Okay. And this callase is produced by the tapetum or the tapetal cells. See, here the microspores are forming. Here the cells are getting rounded off. Callose wall is forming here. Tapetum is very close to it. Once microsporogenesis happens, this will secrete callase that will break down the walls and the microspores will separate. Once the microspores are separated, eventually it will give rise to the pollen grains. Now look here. These cells are giving rise to microspores. That's why you're calling it microspore mother cell. Eventually, microspores are giving rise to pollen grains. So ultimately, it is through these cells that the pollen grains are also forming. So this is also called pollen mother cell. Microspore mother cell, pollen mother cell mean the same thing. Okay. Microspore mother cell in short is called MMC. Pollen mother cell in short is called PMC. Now with all the details that we know, let's try to redefine microsporogenesis. It's of course a process that occurs within the anther where the diploid microspore mother cells or pollen mother cells undergo meiotic division to give rise to 4 haploid microspores. This process is called microsporogenesis. So in the story of our hero, we finished the first part, the sporophyte forming microspores. which is called Microsporogenesis, is over. Now we have to learn about, this part is done. We have to learn about how the microspores develop into the male gametophytes. Okay. See, one microspore mother cell gave rise to 4 microspores. All of the other cells also will produce 4 microspores each. So from each microsporangium, you will end up getting many microspores. So if you look at this anther lobe, I told you that this is this cavity, this chamber, right? The entire inside of it will be filled with sporogenous tissue. So all of the microspore mother cells undergoing meiosis will give rise to many number of pollen grains being produced. Okay. Now, let's talk about how the microspore develops into the male gametophyte. What is the male gametophyte? The male gametophyte is nothing but our pollen grain. A lot of students get confused. They think that the pollen grain itself is the gamete. The pollen grain is not the gamete. The pollen grain is the gametophyte. Okay. So, it is within the pollen grain that the male gametes will form. You know how in angiosperms the major plant body or the dominant phase is the sporophytic phase. The gametophyte is kind of dependent on the sporophyte, does not have independent existence. We've learnt about all of that. The gametophyte is just very few celled. The highest number of cells a pollen grain can have is 3. Okay. So it is a few celled gametophytic phase which depends on the sporophyte. Now, the pollen grain is the male gametophyte. Here you can see there is so much variety and beauty in pollen grains. Pollen grains come in so many different shapes, colors and sizes and various ornamentations. It's very pretty to look at under the microscope. So here you can see a couple of them. Of course, its shape varies. You can see how this is somewhat oval shaped and this one is spherical and this has a weird triangular whatever tubular shape. So its shape varies, but in a vast majority of them the shape is spherical. So you can say that the pollen grains are generally spherical, not always, generally in most cases spherical. Of course, its size is also variable. They're usually microscopic, very small, and it ranges between 25 to 50 microns in diameter. Okay, so that is your pollen grains and its general characters of sorts. We'll see how pollen grains form. See, I told you that the microspores will give rise to the pollen grains, right? So, how is a microspore different from a pollen grain? Let's understand that. We'll take one microspore that we got from Microsporogenesis. So, this is one single microspore which is haploid. This has a wall, a cell wall, like most plant cells. All plant cells have cell walls. So, this one also has a cell wall. And the cell wall here is made up of cellulose and pectin, as in most cases. Now, as this microspore transforms into a pollen grain, as it transforms into a pollen grain, okay, so this wall will remain. This is the pollen grain. Let me write it. There's just one haploid nucleus inside. That hasn't changed. Now this develops a wall around itself. I may have to move this a little bit. I don't think I have sufficient space on this slide. Ouch. Just a minute. Just 1 minute. I'll redo. Yeah. So let's say this is the pollen grain, which is haploid. In addition to having this wall with cellulose and pectin, additionally, it will develop another wall outside it. Okay. Now, this is something that is present only in pollen grain, but you don't find it in microspore. This wall. Yeah. So, this is found only in the pollen grain and not in the microspore. This is one difference. Now, further, how does the pollen grain develop? We'll talk about that. This pollen grain, as of now, I told you that it is the male gametophyte. That means it should have the male gamete inside. As of now, it does not have the male gamete. It just has one haploid nucleus. So within this pollen grain, male gametes have to form, right? So it will further develop. Next, you can see that it will undergo one round of mitosis, which is unequal. Normally, after mitosis from one cell, you get two cells which are kind of similar in size. But over here, unequal mitosis happens because an asymmetric spindle forms. As a result, you will end up getting two cells of different sizes, one very large cell and one very small cell. The outer two layers are going to remain the same. Let me just copy it from here. It has two wall layers. I think you're able to make out the difference in color. The inner one is pink and the outer one is green. Okay. So inside that, it will end up having two cells. One large cell, which is called the vegetative cell. And the small cell that is called the generative cell. Now the vegetative cell that is large has a dense cytoplasm and an irregular nucleus, as you can see. That is the larger cell. And the smaller cell is spindle shaped. It also has dense cytoplasm and a prominent nucleus. This kind of floats in the cytoplasm of the vegetative cell. Okay. So now you have a two-celled pollen grain. This pollen grain has two cells. So you call this the two-celled pollen grain. Now talking about this extra wall. This wall is called the exine. Exo means outside. This is the outer layer of the wall. So it's called exine. Exine is made up of a substance of sporopollenin. Exine is a discontinuous layer, which means there are certain regions in the exine where sporopollenin is absent. Like it's absent here, 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 let's say. And the regions where sporopollenin is absent, these regions are called germ pores. Okay. Now there is one speciality about sporopollenin. This sporopollenin is one of the toughest, most resistant organic materials. Or let me just write substance. Most resistant biological substance. What does it mean? It means that even if you expose the exine or the sporopollenin to extremely high temperature, it will not degrade. Give it extremely strong acids or alkali, it will not break down. Any enzyme you treat it with, any hydrolytic enzyme, it will not break down. So that is the level of VIP security that the cells inside the pollen grain are getting. And it's fair because pollen grains are going to produce gametes and gametes are very, very essential for sexual reproduction. So it requires that level of protection and sporopollenin provides that. Apart from that, sporopollenin in the exine is sometimes known to show certain ornamentations like spines and all of that. So this is usually because if it has to be pollinated by a pollinator like an insect or a bird, if it's sticky or pointed like this and has tiny, tiny spines like this, it is easier for the pollen grain to get attached to the surface of the pollinator. So that's why it will bear some spines. Now talking about the germ pores, why are germ pores even there? Germ pores are regions where sporopollenin is not there. So from there, damage can happen to the pollen grain, right? It's not completely protected on the outside because there are germ pores present. The reason germ pores exist is because it is through the germ pores that the gametes will, the pollen tube will form and the gametes will get transferred much later. It requires protection, of course, because it has precious cells, I agree. But if the cells are present within the pollen grain forever, then it's useless. the male gametes have a job to do. They have to go and fertilize the female gamete. If it's stuck inside, has no exit door, then it will be useless, right? It requires protection and also requires a little bit of freedom. So through these germ pores that acts like the exit door, through these germ pores, the male gametes that are formed inside will have an exit route to get into the female reproductive part and bring about fertilization later. Now still, there are two cells in the pollen grain, vegetative cell and generative cell. The gamete has not yet formed. I have not labeled any cell here as the male gamete, right? So, when is the male gamete going to get produced? In the next step, this generative cell is going to undergo mitotic cell division. Not both the cells, only one of the two cells, the generative cell, is going to undergo mitosis. As a result, hey, what's wrong with you? You can see okay. So within the, a pollen grain, the generative cell will undergo mitosis and give rise to two male gametes. The generative cell, the vegetative cell's nucleus remains as it is, and the generative cell will produce two male gametes. In the exine, the germ pores will exist. All of that I'm not going to label again. Just understand that these pores will exist. And inside, this is what is happening. Now here you have one vegetative cell, one in number, male gametes, two in number. So totally within this pollen grain, you have three cells. That is why this stage of pollen grain is called the three-celled stage of pollen grain. This is the mature pollen grain. Okay. Now, this is also a pollen grain, this is also a pollen grain. In majority of the plants, the anther matures and the pollen grain release happens from the parent plant when the pollen grain is in the two-celled stage. When it is in the two-cell stage, in majority of plants, more than 60% of the plants, pollen release will happen. In the remaining plants, in less than about 40% plants, pollen release will happen after the second mitotic division of the generative cell. Now, if it is released at the two-cell stage, does that mean they will not produce gametes? No. After they are released, the second mitotic cell division will happen in the generative cell. They also will eventually reach the three-cell stage. But what you're trying to say here is that when they are released, they are not already fully mature. They are not in the three-cell stage. They are released when they have two cells. Eventually, they will form the three-cell stage too. Okay, so that is the story of our beautiful, beautiful pollen grains. It is also because of the presence of sporopollenin that the pollen grains are preserved very well as fossils. Let me write it down here. Preserved well as fossils because, it doesn't get broken down by anything. So that is the pollen grain. I told you that this is the male gametophyte. Pollen grains are the male gametophytes. Within the pollen grains, male gametes will form. We already saw the formation of male gametes here. So we're moving ahead in the story. So here we have, oh my God, where is that slide? Oops. Hey, did I delete it by mistake? No, it's here. Okay. So we saw how the microspores developed into the male gametophyte, which is nothing but the pollen grain, and we saw how within the pollen grain the male gametes formed by undergoing mitosis. So the hero story is kind of over, but we have a little bit left to learn about pollen grains. We'll finish that and then we'll jump off to the female part. Okay. So about the structure of pollen grain, whatever I've explained, you have it in the slide here in the form of text. You may go through that. Here is an NCERT diagram that's showing how during division of the microspore, you can see an asymmetric spindle formation which will eventually give rise to the vegetative cell that is larger and the generative cell that is smaller. Unequal cell division happens here. And yeah. So pollen grains, about its advantage and disadvantage to us humans, we'll talk about. A lot of pollen grains are released during the flowering season. You know, I told you that in a lot of species of plants, only when the season is favorable do the plants flower. Usually during spring, summer, that is when the flowers are produced. So at that time, a lot of these flowers of a lot of species release the pollen grains into the air. Since they're very, very tiny, dust-like, a lot of humans are kind of hypersensitive to its presence, and the pollen grains can sometimes cause respiratory illness like asthma, bronchitis, and all of that. Okay, so it's it can cause allergies in humans, especially the Parthenium plant, which is also called carrot grass. Actually, this carrot grass is not a native plant to India. This plant is not native to India. That is the correct phrasing, yeah. And we got it through from United States of America, I suppose, when we were trying to import wheat from them very, very long ago. It got into India as a contaminant and it has never left ever since. So, yeah, so this also produces very, very tiny pollen grains, which is known to cause allergies in a lot of people. So, that is one disadvantage of pollen grains to us. Okay, so it causes allergies, bronchial afflictions, asthma, bronchitis, and all of that, especially Parthenium. It causes pollen allergy. What is its advantage to us? Its advantage is that they are believed to be highly nutritious, because of which it is sold in the markets in the form of syrups and tablets which people consume. And it's also claimed that it will increase the performance of race horses and athletes, and they are also known to consume it, which is an advantage. Okay, so it is rich in nutrients, available in the form of tablets and syrups. Athletes and race horses use it to enhance their performance. Moving on, we have to talk about a topic called pollen viability. Now see, this is our anther. Anther has produced pollen grains. Pollen grains have left the anther. Anther broke open and released the pollen grains. And inside the pollen grain, you have the two male gametes lying around. Now this male gamete, what is the job of this male gamete? It has to go and fuse with the female gamete. But where is this female gamete? Female gamete is present inside the gynoecium. We'll learn about its detailed structure later, but for now, for your understanding, I'll show you a little bit. Wait, let me draw it here. Yeah. Okay. So inside the ovary, you will have the ovule, and inside the ovule, here is where the female gamete is present. So this male gamete inside the pollen grain has to go into this ovule and fuse with the female gamete. How does it bring that about? So here outside you have exine, right? Exine has some germ pores. Once it comes and lands here, okay, let's say this is one germ pore. This is the intine, which is cellulosic, which is made up of cellulose and pectin, normal cell wall. Inside this, you have the two male gametes. What happens is, once it comes and falls on the stigma, it has to form a tube. It forms a tube, and through this tube, the two male gametes will enter the ovule. The process by which this pollen grain forms a pollen tube, this is called pollen germination. See, this pollen grain has already left the parent plant. For as long as it was in the parent plant within the anther, it used to get nourishment from the connective. Connective has vascular supply. From there, it gets food. From the tapetum, it's getting nourished. But once it leaves that, it no longer has access to additional food or nourishment. So whatever reserve food is present in the vegetative cell, for as long as that is available, this pollen grain can survive. But beyond that, it will not be able to survive. Once it has very little nutrients available, it will lose the ability to form this pollen tube. It cannot germinate any longer. So once it has lost that ability, we say that the pollen is not viable anymore. The period during which it retains the ability to form that tube is called pollen viability. Now, how long the pollen viability will be for a particular species varies based on a lot of factors. It varies from species to species. For example, if you consider the grass family, Poaceae members like rice and wheat, after about 30 minutes after the pollen grains have been released, it loses its viability. But in some families like Leguminosae, Solanaceae and Rosaceae, you will notice that the viability period is kind of extended for a couple of months. It will retain the viability period. So that also varies on a lot of environmental factors like temperature and humidity. Whenever there is high temperature, the metabolic activity inside the pollen grain also will get increased and the nutrients will get exhausted much sooner. And humidity. When there is a lot of moisture in the atmosphere, then the pollen tube germination will happen kind of prematurely. That can cause pollen germination, it can cause microbial infection within the pollen grain, all of that can happen. So it depends on these external factors as well. So if you think about plants like rice and wheat, you know, its pollen viability is very, very short, like half an hour. 30 minutes, that's all, right? If the pollen viability period has to be extended for artificial hybridization or any plant breeding methods, then it can be done by storing these pollen grains at extremely low temperatures as low as minus 196 degrees Celsius in liquid nitrogen. So that way you can extend the pollen viability period, and these are called pollen banks, and storing it at minus 196 degrees Celsius in liquid nitrogen is also called cryopreservation. Okay, another point that I forgot to mention is there is one question in your NCERT exercise uh, where they're asking about how is the sporopollenin obtained from? Where do you get sporopollenin from? Is the cell itself producing this extra wall? See, I told you here, this does not have a wall, this has a wall. Now, where did this wall come from? Any wall will need raw materials. If a wall has to be built, you need bricks for that, right? So, the precursors for sporopollenin wall, did that come from inside the cell or from outside the cell is the question. Whenever a cell layer, a wall layer is formed inside the cell towards the plasma membrane, towards the cytoplasm, then the substances or the raw materials or the bricks required for the wall formation comes from inside the cell. Over here, the sporopollenin wall is forming outside. So how is this sporopollenin wall forming? Where is the raw materials for that wall coming from? So that is coming from tapetum. Look here, I told you that the tapetum is lying very close to the sporogenous tissue, right? So once microspores form here, while they're forming pollen grains, while they're transforming into pollen grains, it is the same tapetal cells that used to nourish the microspores that will start producing and releasing the precursors required for the sporopollenin wall formation. So the sporopollenin is formed from the tapetum and not from the pollen grain itself. Another thing about tapetum is that sometimes it produces certain substances that will make the pollen grain sticky. It's important for pollen grains to be sticky in insect pollinated plants, because if it's not sticky and too dry, it will not stick on to the surface of the insect's body or the bird's body, any animal pollinator's body. So for making the pollen grains surface sticky, tapetum is responsible again. Okay, tapetum nourishes the developing microspores. It gives and provides the raw material required for sporopollenin formation. It also makes sure that this pollen grain surface becomes sticky so that it can attach to the body of the pollinator. So that's about our beautiful hero, pollen grain. With that, we have finished one half of the story. Was it as dramatic as the hero that you see in movies while their intro scene is like either they're playing basketball or they're like punching somebody in the face, right? So this is a more decent hero in flowering plants. So next we'll talk about the female lead, which is the gynoecium. The gynoecium is also called the pistil. Similar to how androecium had many units that we call stamens, in a gynoecium you will have units that are called carpels. Gynoecium is the female reproductive part of the flower, and the number of carpels in a flower varies from species to species. Sometimes each flower will have only one carpel, in which case we call it monocarpellary. Mono means one. In some flowers you have many carpels within the same flower, and there you call it multicarpellary. Multi means many, that means many carpels are present. When many carpels are present, the carpels may be fused to each other or they may be free. When they are free, you call it apocarpus, where the carpels are free. When the carpels are fused, you call it syncarpus. Examples for both of these, it's important for you to remember. Apocarpus example is Michaelia. This is called Sampiga in Kannada. I don't know what it's called in other languages. Michaelia champaka. Champaka, if you know, it's a very fragrant, beautiful yellowish orange petal having flower. That flower has Apocarpus ovary. And Papaver somniferum. Papaver is this poppy plant. In this plant, you will have many carpels that are fused. So remember this example too. Now we have to talk about the structure of gynoecium. Gynoecium typically has three parts. It has a stigma, which is the first part. Then it has a long, slender structure called the style. And at the base, it has a broad swollen structure that is called the ovary. Now, when pollen grains have to fertilize the egg, which is lying inside here in this ovule, it has to come and land on the stigma itself. Here is where it can form the pollen tube. If it comes and directly falls here, it will not be able to form the pollen tube. So this is like the entrance for the pollen grain into the female reproductive part. So this is like the landing platform for pollen grains. In order to trap the pollen grains, this stigmatic surface can be a little sticky. It will also have a lot of sugars that will support and nourish the pollen grain and allow it to form the pollen tube. Next, you have the style. It is quite boring. It is just connecting the stigma to the ovary. Next, you have the ovary. Ovary is the broad basal bulged part of the pistil. Within the ovary, you will have many ovules present. Okay, so these are the ovules. It is within the ovules that the female gamete will form. Now, if you look at the ovary, which is the broad part, you will notice that within the ovary, you can find a thick fleshy structure like this, which is called the placenta. Placenta need not always be in the center. It can be in different positions. That also changes from species to species. You learnt about placentation in the previous year. I hope you remember. What type of placentation is this? This is axial placentation. To a central axis, ovules are attached, right? To this, the ovules will be attached. So these are the ovules. Inside the ovary, whatever cavity is there, that is called the locule. So this space here, that is the locule. So in one ovary, one to many ovules can be present. In some species like grass and wheat, each ovary carries only one ovule. But in some species like peas, beans and all of that, within one ovary you can find many ovules. Their number varies. Okay. So wheat, paddy, mango has only one ovule per ovary. In papaya, watermelon, orchids, you have many ovules. How does one know if the ovary has one ovule or many ovules? Eventually, after fertilization, the ovary wall is going to become the fruit wall and the ovule is going to become the seed. If an ovary or if a fruit has many seeds, that means the ovary had many ovules. If a fruit had only one seed, that means the ovary has only one ovule. In mango, you see only one seed, but in watermelon, you see many seeds, right? So that's one way you can remember the examples. Now, similar to how we learnt about the structure of stamen first, we learnt about the structure of gynoecium here. What did we learn after stamen? We learnt about the structure of microsporangium. Here again, we have to learn about the structure of megasporangium. Now, where is the megasporangium located? It's located inside the ovary. Now, it is within the ovule that the megaspore will form. If this is a structure that will give rise to the megaspore, what should we call this structure? Ovules are also called megasporangia. Previously, the structure that gave rise to microspores, we called it microsporangia. Now, the ovule is going to give rise to the megaspore. So, ovule's alternate name is also called megasporangium. You know, in leaves there are stalks. In some flowers, you have stalks, right? The stalk of the leaf is called a petiole. The stalk of the flower is called a pedicel. We learnt about this just today. Like that, even the ovary will have a stalk. if this is the stalk of the, I'm sorry, not the ovary, the ovule. This is the stalk of the ovule. Let's say this is the body of the ovule. The stalk of the ovule is called the funicle. Depending upon the position of the stalk or the funicle with respect to, there will be a small opening present at the tip of the ovule called micropyle. Depending upon how they are aligned with respect to each other, you have many different types of ovules in angiosperms. In this you can see, this is the ovule. It's not clear, let me use a darker color. This is the ovule, this is the stalk. The stalk and the micropyle are in the same line. But if you look at this is the stalk, this is the micropyle. Both are parallel to each other. If you see here, this is perpendicular. So, different types of ovules are present. Here, in angiosperms, we are going to focus on anatropous ovule because in most angiosperms, this is the type of ovule that you will find. Now we are going to learn about the structure of this anatropous ovule. Now this anatropous ovule is an inverted ovule. What happens here is the ovule is supposed to be like this with this pore. This becomes ultafied. This is normal. This is orthotropous. But in inverted ovule what happens? This is the stalk. This body of the ovule gets inverted like this. Okay. So you will see that the body of the ovule at one point is fused with the stalk. Okay. So we learn about that now. Megasporangium is also called the ovule. Keep that in mind. Similar to how Microsporangia had a wall that had many layers that were offering protection, here even in the Megasporangium, since megaspores are being produced, they also are precious cells, they need protection. The ovule has two integuments that are protective in function, two layers. Outer one is called outer integument. And the inner layer is called the inner integument. Since this is an inverted ovule, you do not find the stalk here. Okay. The stalk is here. And this stalk is called the funicle. You can see that this is the body of the ovule. The body of the ovule is fused with the stalk of the ovule at this position. We call that the hilum. These integuments offer protection, but it does not completely cover what is lying inside because eventually a tube has to form, bringing the pollen grains inside. So there should be an entry point inside the ovule. So the integuments leave a small space in between, which is called the micropyle. What is present inside? Inside you can find a homogeneous mass of cells called nucellus, not nucleus, nucellus. Okay, so this is the nucellus. Within the nucellus, you will find many cells present. Okay. Now the end of the ovule where the micropyle is present, this end is called the micropylar end. And the opposite end is called the chalazal end. The chalazal end is called the basal part of the ovule. Why is that so? Because here you can see if this is the funicle, this is the micropyle, this is the chalaza. So this is like the base of the body of the ovule. Right now, it does not seem like the base because the ovule is inverted. But if you look at the body of the ovule, this itself is the base, right? So this is called the basal end. This is the structure of our Megasporangium or the ovule. Now, our next job, similar to how after studying the structure of Microsporangium, we learnt about Microsporogenesis. Now, we've discussed Megasporangium structure. Next, we have to learn about Megasporogenesis. It is the process by which megaspores form. Okay, so we'll talk about Megasporogenesis. There are a few differences between Microsporogenesis and Megasporogenesis about which I will talk now. So, since megaspore is formed from the nucellar cells, I'm only showing the nucellar tissue here. Okay, so this is the nucellus. Within the nucellus, you have many cells present. So let's say this was the micropylar. This is the micropylar end, and this is the chalazal end. What happens is, one of the cells from the chalazal end will get differentiated. It will grow larger in size compared to the other cells. Okay, so the other cells are kind of small. This one specifically towards the chalazal end will grow slightly larger. It will have a dense cytoplasm and prominent nucleus. You can clearly tell that this cell is different from the other cells by looking at it under the microscope. Now, this unusually large cell here is called the megaspore mother cell. Earlier, you called it the microspore mother cell. Now, you're calling this the megaspore mother cell. The megaspore mother cell is diploid because it's a part of the sporophytic plant body now. This one megaspore mother cell will undergo meiosis to give rise to megaspores. Okay, so now we'll take the story of only one megaspore mother cell. This is one diploid megaspore mother cell. If it's megaspore's mother, then obviously it has to form the megaspore. During spore formation, what kind of cell division will take place? Meiotic cell division will take place. As a result of meiosis, you end up getting 1, 2, 3, 4 haploid megaspores. Earlier, I told you when microsporogenesis happened, we spoke about one microspore mother cell ending up to form 4 microspores. But I mentioned whatever is the story of this microspore mother cell, all other microspore mother cells also will end up having the same fate. But that is not the case in megasporogenesis. In megasporogenesis, you will notice that only one cell will differentiate to become the megaspore mother cell. The other cells will not undergo meiotic division. Among all the nucellar cells, only one cell is going to undergo meiotic division and give rise to 4 megaspores. Also, among these 4 megaspores, 3 are going to degenerate. Only one will remain functional. And this functional megaspore will give rise to the female gametophyte. What is the job of the spore? To form the gametophyte. This will form the female gametophyte. Okay. Since for the formation of female gametophyte, only one megaspore is involved, This development is said to be monosporic. Mono means one spore is involved. So it's called monosporic development. Okay, what happens to the other nucellar cells? They kind of offer nourishment to the megaspore mother cell for megasporogenesis. They will support the development of the embryo sac and eventually they will also degenerate. Okay, now how would you define megasporogenesis? Megasporo definitely relates to megaspore. Genesis means production. So megasporogenesis is a process by which diploid megaspore mother cells undergo meiotic division to give rise to haploid megaspores. Where does this take place? This takes place inside the Megasporangium or the ovule. Now, how does this megaspore develop into the female gametophyte is what we have to look at. It is very, very easy. Okay, so our functional megaspore is yellow color. Let us continue with yellow. Here we have our beautiful haploid functional megaspore. Remember, when the megaspore forms the gametophyte, what kind of cell division takes place? Mitosis takes place. In male gametophyte development from the microspore, we saw that first an unequal mitotic division happens and a larger vegetative cell and a smaller generative cell forms, and the second mitotic division only the generative cell will undergo. So there are 2 rounds of mitotic divisions. Over here, during the formation of female gametophyte, 3 rounds of mitotic divisions will take place. Let's see how it goes. So here, after the first round of mitosis, I'll just write MIT, understand that it is mitosis. 2 nuclei will form. See, every time a cell divides, if this is one cell, first after S phase it will make an extra copy of its DNA, and after karyokinesis the nuclei will separate, and after cytokinesis between the 2 nuclei a cell wall forms and the 2 cells separate. So this is called karyokinesis, that is the division of the nucleus. And oh my god, why is the fan on? Just a minute. Yeah. The 2 nuclei will form. I was here, right? Yeah. So this is called cytokinesis. It's a single word, but there's no space, so I'm writing it as 2. Here what happens during mitotic divisions is, it is free nuclear, meaning karyokinesis will occur, but cytokinesis does not immediately take place. It's not like it will never happen, but right now it is not happening. So after the first mitotic division, you end up with a single cell having 2 nuclei. We can call this the 2 nucleate stage. Now, after the 2 nuclei are formed, these 2 nuclei will migrate towards the opposite poles, and at the poles they will undergo another round of mitosis, again where karyokinesis takes place, but cytokinesis doesn't immediately follow. So what do we end up having? After these move towards the opposite poles, you end up having 1 nuclei at each pole. Now that each nucleus is undergoing mitosis the second round, you will end up having 4 nuclei, 2 at each pole. This is called the 4 nucleate stage. After that, it undergoes another round of mitosis, the third round of mitosis. Again, both these nuclei, all the 4 nuclei will undergo another round of mitosis where karyokinesis happens, but cytokinesis doesn't. So you end up having, not you, like this structure ends up having 1, 2, 3, 4, 1, 2, 3, 4, 4 nuclei at each pole. This is called the 8 nucleate stage. Now in the next step, mitosis does not take place, mitosis takes place only thrice. Over here, finally, cytokinesis will happen, but before that, some reorganization will happen. Reorganization along with cytokinesis. So what happens during reorganization? one nucleus from each pole will migrate towards the center. If one nucleus from each pole migrates towards the center, you will have 3 nuclei at each pole and 2 nuclei at the center, right? Total 8 nuclei. Now, once this reorganization happens, between these nuclei, cell walls will form. And this is our mature embryo sac. In our mature embryo sac, you have a total of 1, 2, 3, 4, 5, 6. This entire thing is one cell, 7. So you have a total of 7 cells. How many nuclei do we have? There is no change in the number of nuclei. There were 8 nuclei here. There's going to be 8 nuclei here too. So 8 nuclei. In between these two nuclei, a cell wall will not form. In between the other nuclei, cell walls will form, but over here it will not form. So in this huge cell at the center, there are 2 nuclei present. That is our 7-celled, 8-nucleate embryo sac. Okay, so the female gametophyte is also called embryo sac. I don't think I mentioned that before. Another name for the female gametophyte is the embryo sac, because it is in this structure that the embryo forms in the future. Okay. Now moving on, I'll tell you about what are the different cells in this embryo sac called. Okay. First, let's draw one koli motte. This is a nice shape, but not a nice color. We'll use a better color. We had 2 nuclei present at one end, and I'm sorry, not 2 nuclei, 3 cells present at one end, 3 cells present at the opposite end. Each of these cells have 1 haploid nuclei, and this large cell at the center has 2 nuclei. Now, this is forming where? This is forming inside the ovule. Here is one cell, megaspore mother cell, differentiated here towards the chalazal end. That is what gave rise to the megaspore, the functional megaspore, of which 3 degenerated, 1 remained functional. That functional megaspore existed right here, and that functional megaspore gave rise to the embryo sac right here. So here is where your embryo sac form. Okay, so here you have ayayyo. 1 minute. I'll use another technique to color this. So here is where whatever structure we are talking about that forms inside the ovule, right here. Previously we saw that the male gametophyte pollen grain was released from the anther, but the female spore or the gametophyte will not be released from the ovule. Okay, so it will be retained within the ovule. Our embryo sac is right here within the ovule. Similar to how the ovule has its chalazal end and the micropylar end, the embryo sac also will have its chalazal end and micropylar end, right? So towards the micropylar end of the embryo sac, this side, you have, or you can even call it the micropylar pole. So you have 3 cells. Collectively, these 3 cells are called the egg apparatus. In an egg apparatus, you will have obviously the egg cell. This central cell here is the egg cell. This is 1 in number. On either sides of the egg cell, the 2 cells that you find here, this one and this, wait. How do I show this? Okay, So this one and this one, the one on the sides, these are called synergids. They are 2 in number. All of them are haploid because this is a gametophyte and gametophytes are haploid. The egg cell is haploid and the synergids are also haploid. Now there's one special feature that synergids have. Towards the micropylar pole, these synergids will produce or will have finger-like cellulosic thickenings, the cell wall thickenings. That is called the filiform apparatus. Okay. So that is your egg apparatus. Towards the chalazal end here, you have 3 cells. And these 3 cells together constitute what is called the antipodals. Antipodals are 3 in number and they are haploid. This large cell at the center is rightly called the central cell. The central cell has 2 nuclei, both of which migrated from the poles on opposite sides. So these 2 nuclei here are called the polar nuclei. Since there are 2 haploid nuclei here, does that make the central cell diploid? The central cell is not diploid, it is still haploid, but it is multinucleate. When can you say that a cell is diploid? You can say that a cell is diploid when you have 2 haploid nuclei that have fused. Okay, so over here, the nuclei are not fused. There are 2 separate nuclei, both are haploid. So that makes the central cell multinucleate and haploid, it's not diploid. So if you take account of the number of cells here, you have 3 antipodals, 1 central cell, 1 egg cell, 2 synergids, 2 plus 1, 3, 3 plus 1, 4, 4 plus 3, 7. Total number of cells is 7. So here you have 7 cells. All the cells apart from the central cell has 1 nucleus each. The central cell has 2 nuclei. So totally you have 8 nuclei. Every one of the cells in the female gametophyte is haploid. Even in the male gametophyte, all the cells are haploid. The 2 male gametes, vegetative cell, generative cell, everything is haploid. Everything in a gametophyte in any plant is always haploid. Don't get confused with the ploidy. Synergids, egg cell, antipodals, everything is haploid. Okay, now we learned about the development of the female gametophyte. Here, I have to tell you one thing. In some diagrams, they give you the egg apparatus on top and the antipodals at the bottom. So, does that make any difference? Why is it at the base at sometimes and sometimes on the top? It doesn't matter whether the egg apparatus is on top or at the bottom. All that matters is egg apparatus has to be at the micropylar end, antipodals have to be at the chalazal end. If egg apparatus is here, that means this is the micropylar end. That's how it is. Okay, so that's about your female gametophyte. Now let's move on and talk about, alright, so here's a difference between microsporogenesis and megasporogenesis, which you can take a look at in your notes. I will provide this slide. And moving on, let's talk about pollination. Right now, what all has happened? Let's take a quick look at that before we move on to pollination. Okay, are you? This is one Tamil song that's stuck in my head, okay. I just won't. Okay, it's here. So we spoke about megasporogenesis here. This process, we just now discussed about that. It happens in the ovule. Megaspores are formed, right? From the megaspores, female gametophyte or embryo sac formed. Within the embryo sac, you saw that the female gamete or the egg cell was produced. The egg cell here itself is the female gamete. Inside the pollen grain, 2 male gametes are waiting. Within the embryo sac, there is 1 female gamete or the egg cell waiting. Now the thing is, this male gamete has to come in contact with this female gamete so that fertilization can happen and the zygote can form. So that is our next step. Where are you now? So here you had the anther. Anther produced the pollen grains. Anther dehisced and the pollen grains were released. The pollen grains are now out in the open. Within the pollen grains, you have two male gametes. Of course, there's the vegetative cell too, but I'm not choosing to label it right now. And then inside the ovary, you have the ovules, and inside the ovules, you have the embryo sac, and of course, inside that, you have the egg cell. For this male gamete to go and fuse with this egg cell, this has to be transported from the anther to the stigma. Right? Pollen grains are non-motile. They do not have cilia, flagella, legs to move around and go sit on the stigma. So, certain agents are required to transport pollen grains from the anther to the stigma. We'll talk about that later. Also, this process is called pollination. The process by which the pollen grains that are produced where? By the anther are transported to where? To the stigma. That process is called pollination. It is the transfer of pollen grains that are shed from the anther to the stigma of a pistil. Now, this pollination is of three major types. You have autogamy, gatonogamy, and xenogamy. Let's learn about each one of them. Now, if you're asking me what is the basis for this classification, the basis for this classification is the source of pollen grains. Depending upon whether the pollen grain that is getting pollinated belongs to the same flower as the stigma, or is it from a different flower but the same plant, or is it from a different plant altogether, you classify it into autogamy, gatonogamy, and xenogamy. Let's start with autogamy. Auto means self, so this is self-pollination. In autogamy, what happens is, you have a flower. This flower is typically bisexual. So it has the anther and it also has the pistil, right? So the pollen grain that this flower produces, if it lands on the stigma of the same flower, then you call it autogamy. For autogamy to take place, the flower has to be bisexual. If it's not bisexual, it's impossible. Also, the anther and the stigma have to be very close to each other. In autogamy, you will notice that only one plant is involved, and within that plant, only one flower is involved. Okay? 100% it has to be bisexual, only then can autogamy or self-pollination take place. In nature, if you look at, there are many species of plants that produce two different types of flowers. For example, there's viola, camelina, oxalis. These plants, they produce two different types of flowers. What are the two types of flowers? One is chasmogamous flowers. These are regular flowers which bloom, and when they bloom, the petals open and it exposes the anther and the stigma. So these are open flowers. And then the other type of flower is called cleistogamous flowers. So these cleistogamous flowers, they are closed, meaning they don't bloom at all. The anther and the stigma are never exposed to the outside. For example, if you look at this plant, this is camelina plant. Aerially, if this is like the soil surface, aerially it produces chasmogamous flowers that are open, that bloom and expose the anther and stigma. But underneath, like within the soil, it produces certain flowers that are cleistogamous, which means they don't open at all. Same plant producing two different types of flowers. These are closed. Now, in chasmogamous flowers, even if it is bisexual, you can't always ensure that autogamy will take place. Autogamy or gatonogamy, self-pollination or cross-pollination, either of them can take place because the anther and stigma are exposed. But in case of cleistogamous flowers, since they don't open at all, imagine you have a flower where the petals are not opening at all. anther and stigma are forced very close to each other inside the bud. Since there is no exit or entry into the flower, pollen grains that are produced inside the flower have no escape. They can't go and fall on another flower stigma. And pollen from another flower stigma cannot enter inside because it's closed from everywhere, from every side. So in such flowers that are cleistogamous, you can't expect cross-pollination to happen 100% of the time. Invariably, it's going to be self-pollinated or autogamous. So in cleistogamous flowers, you can see autogamy being invariable. Invariable means there is no change. There is 100% of the time autogamy taking place. Now, there is advantage and disadvantage in autogamy. The advantage is that you don't need external pollinating agents for pollination to take place. Since they are present so close to each other, the anther and the stigma, as soon as the anther matures and releases the pollen grains, it's going to land directly on top of the stigma, and pollination will happen, and fertilization will happen. If fertilization happens, then obviously seed formation will eventually happen. Even if there is no external pollinating agent like wind or water or animals or nothing, even without pollinating agents, they will have seed formation assured. Guarantee seed production, you can see there. That means 100% they're going to make babies. What is the disadvantage though? Can you think about it? Continuously, if self-pollination is happening, then the variation will reduce in that population, which is not so great for the health of that population or species. So that is one disadvantage. Okay. Now another thing, in cleistogamous flowers or in flowers where autogamy has to happen, we also have to ensure that whenever the anther matures and releases the pollen grains, at around the same time the stigma also has to be receptive. If the stigma is not receptive but the anther is mature and releases pollen grains, it cannot undergo autogamy. Okay, so if autogamy has to happen, there has to be synchrony. Synchrony means it's happening at the same time, simultaneously. Okay, and another thing, the anther and the stigma must lie close to each other. Moving on, let's talk about gatonogamy. Yeah. So gatonogamy involves, the flower could in this case be either unisexual or bisexual. Previously, I told you that the flower has to be 100% bisexual, otherwise self-pollination is not possible. But in gatonogamy, even if it is unisexual, it's possible. In gatonogamy, what happens is, you have one plant within the same plant, you have two different flowers. Okay, so let's say this is one flower and here you have another flower. They may or may not be bisexual, but just for convenience, I will draw a bisexual flower. This has its anther, this has its own anther, and both of them obviously will have their pistil as well. So the pollen grains that are produced by one flower, if it lands on the stigma of another flower belonging to the same plant, then you call it gatonogamy. Over here, you have one plant involved, but two flowers of the same plant. Previously, one plant, one flower. Now, one plant, two flowers are involved within the same plant. Though, if you're looking at it functionally, pollination is happening between two flowers. So, functionally, you can think of it as cross-pollination. But if you think about it, since both of the flowers are from the same plant, whatever gene combinations this has, this will also have the same gene combinations because it's from the same plant, right? So, it's not genetically cross-pollination. Genetically, it is similar to self-pollination or autogamy, but functionally, it is cross-pollination. Over here, transfer of pollen grains happens between the anther and the stigma of another flower, but the same plant. I hope it's clear. Now moving on, let's talk about xenogamy. This is the only type of proper functional cross-pollination. Here, again, the flower may be unisexual or bisexual. and there are 2 plants involved and 2 flowers. So here we have 2 different plants. Each one produces a flower. This one has its anther. Even if it's bisexual, it's possible. I'm sure you understand that. Here you have the female reproductive part. When the pollen grains produced by this lands on this and the pollen grains produced by this lands on this, you call it xenogamy. This is functional and genetically cross-pollination. This is the only type of pollination in which you can expect pollen grains having different genes coming in contact with the pistil. Okay? So this is the only type of pollination during which pollen brings genetically different types of pollen grains to the stigma. So that's about how you have 3 different types of pollination based on the source of pollen. In autogamy, same flower pollen lands on the stigma of the same flower. In geitonogamy, pollen from one flower lands on the stigma of another flower belonging to the same plant. And then in xenogamy, you have pollen from one flower landing on the stigma of another flower belonging to a different plant. Now let's talk about the agents of pollination. If at all cross-pollination has to happen, if self-pollination is happening, then the distance that the pollen grain has to travel is not too much. If cross-pollination is happening, like I mentioned earlier, pollen grains do not have locomotory structure, so there is a reliance on external agency for the transfer of pollen grains. So these agencies that will transfer pollen grains from the anther to the stigma can be living or non-living. In case they are non-living, you call that abiotic agents. It's nothing but wind and water. If it's living, obviously animals, then you call it biotic agents. In nature, if you look at angiosperm species, in a vast majority of angiosperms, pollination happens by means of animals itself. Compared to biotic, abiotic is fewer. And within abiotic, if you have to compare between wind and water pollination, wind is more common compared to water pollination. So among the 3, top is by animals, next by wind, least number of species get pollinated by water. Okay, here you know how pollen grains coming in contact with the stigma is a chance phenomenon, right? So large number of pollen grains are produced. The number of pollen grains produced compared to the number of ovules in a plant is very drastically different. Similar to how in humans you see the sperms that are produced are in thousands, I suppose. But every cycle females produce only 1 egg. In most cases, rarely 2 or 3 may be produced, but that's a rare phenomenon. Likewise, even over here, compared to the ovule number, the number of pollen grains that are released are numerous. Okay, that's because if only 1 or 2 are released, the likelihood of it going and falling on the stigma, bringing about fertilization is very less. To increase the chances of successful pollination and fertilization, the number of pollen grains produced is in very large numbers. Now let's talk about individual types of pollination, agents of pollination, starting with wind. Okay, so wind pollination is also called anemophily. We have to learn about how, if a plant is undergoing wind pollination, how are its flowers adapted accordingly? What are the characteristics that you can find in a flower which is going to undergo wind pollination? Now, if you think about it, if you look at the structures of a flower, the sepals, petals and all of that, you know that petals exist for a reason. It is meant to attract pollinators. Petals being so colorful and showy and large is because an insect or a bird, if it looks at it, it should be attracted and drawn towards it. But here, the pollinating agent does not have life. It is wind. Wind cannot see, wind cannot smell, wind cannot taste anything. So it's pointless for flowers that are pollinated by wind to be bright and showy and put on a lot of makeup because the wind cannot see anyway. There's no point in these flowers emitting fragrance because the wind cannot sense it. There's no point in these plants and flowers producing a nectar because the wind cannot drink it, right? It cannot consume it. So the flowers here are not very showy, they're not very brightly colored, they're very, very tiny, very small in size. The petals are also highly reduced. In some cases, they don't even have sepals and petals separately. They have what is called perianth, a single non-accessory whorl. So it's highly reduced. If you look at it, it doesn't even come across like a flower or what you typically associate with a flower. So they are reduced. Individual flower is very small in size. And many flowers come together and cluster and form an inflorescence. This can be most notably observed in the case of grasses. If you have ever taken a look at the inflorescence of rice or wheat or maize, ragi, any of these, you will notice how the flowers are not very bright there. Okay, because in becoming bright and putting on that makeup also energy is required. Why should it waste energy for something like wind? It is futile, right? It's useless. So here you don't see such bright showy flowers. Now the flowers are tiny here and they're packed into an inflorescence. How should the stamen be? How should the pollen grains be? How is the stigma going to be? This I will tell you. First, let's talk about stamen. I told you that cross-pollination is always advantageous to the plant compared to self-pollination, because when cross-pollination happens, newer gene-containing pollen grains can come and fall on the stigma, and it can increase variation, right? So that is essential for a population. So here what happens is, in order for pollen grains to be in order for the stigma, the anther, to release pollen grains outside so that cross-pollination can happen, the anther has to be very well exposed. So if this is like the non-essential whorls in the flower, let's say, the anther is not hiding inside, it's not small here. The anther is exposed. Exposed means it's come out of the non-essential whorls. Okay, so that when the anther dehisces and releases the pollen grains, it can be dispersed to long distances by air currents, so wind currents, whatever. The stamen is well exposed so that pollen can be dispersed easily by wind currents. How should the pollen grains be? Understand that the pollen grains that are produced have to be carried by wind over long distances for successful cross-pollination to happen. If this pollen grain is one big fat fellow, very dense, it's going to get dropped off very close to where it was produced because of gravity. You do not want that to happen. So these pollen grains are light in weight. Okay, so pollen grains are light here. Light here is not light in color. It's not light colored. It is light in weight. And they are non-sticky. If they are sticky, that's another problem again. If they have a sticky surface, then it will stick on to random surfaces which you do not want. So that's why it is non-sticky. But if it is non-sticky, then how does it stick on to the stigma? How can it possibly stick on to the stigma? Doesn't it get, carried away by wind? There is a trap here. The stigma here is modified in such a way that it will trap the tiny, tiny non-sticky pollen grains. See here, talking about the pollen grains, they are obviously large in amount, light in weight and non-sticky. The stigma here is like a feather. Okay, it's like this. When you want to clean dust at home, we use feathery stuff, right? You don't use a cloth to take out dust. If it's very, very minute dust particles, your broom, your whatever dusters you have at home to remove the cobweb and all of that, that usually has bristle-like structures. That's because when the structure is like that, it tends to trap tiny, tiny dust particles easily. Here, if the stigma were to be flat like this, or if it were to be rounded like this, then this wouldn't stand a chance to land here. It'll just get carried off by wind because it's not sticky. Because this is feathery, as the wind is blowing, as the wind currents are passing through this feathery stigma, this tends to get trapped here. Okay, so that is how the pollination process is, tried to be more successful. So here the stigma is large and feathery so that it can trap the airborne pollen very easily. And here in this case, each ovule will have one ovary. did I say it ulta? Each ovary will have one ovule only. You can see that in the case of maize. You can see that in the case of wheat, in rice, and all of these plants. Usually in grasses, wind pollination happens. So that's why the flowers are not very showy. Okay, not very pretty that you can use to decorate your homes or anything. Now, in maize, everybody must have eaten corn in your life. So, you know, when you peel off the green color outer layers in a corn cob or in the ear of the corn, you know, there are these very nice, silky, shiny hair-like things that arise. That silky hair is called the corn silk, okay? So, the corn silk here is nothing but the style and the stigma that is meant to trap pollen grains, okay? So, if you look at, see, I'll tell you what a year is, what a cob is and all of that. So here you're looking at the ear of the corn. Ear of the corn includes the outer green color layer, inner of these grains of maize, and inside that solid rod-like part. All of that is included in the ear of the corn. Now what is a cob? Cob is the central part. After eating all of these outside stuff, you throw out that central hard part, right? That cylindrical part, that is the corn cob. Now in maize, you will notice that there are two different types of inflorescence, the male inflorescence and the female inflorescence. Here you have the male inflorescence at the tip, at the apex. The male inflorescence is called the tassel. Here towards the axils, you will have the female inflorescence. The female inflorescence is called the ear, okay. Male inflorescence will have only male flowers, female inflorescence will have only the female flowers. If you look at the male flowers, I want you to notice how the anthers are. Anthers are here. So these anthers are exposed, very well exposed, so that the pollen grains when they are released can be easily carried. Now look at the female flower. This is one single female flower. I want you to notice how this is the ovary, this is the style, and this is the stigma, and it is feathery. When the stigma is feathery, it tends to trap pollen grains very easily. To give you like a real life picture, all of these hair-like structures that you're looking at, that is the style and stigma combined. Okay. So if you look very closely at the hair of corn or maize, you will notice that this is one long hair. Another thing, from each of these, one hair would have arisen. The ones towards the tip will be shorter, the ones from the base will be much longer. It's going to be like that. So for the most part, it's going to be smooth, but towards the tip, each hair will have feather-like structure. On my computer, I could see this clearly, but here it is not very evident. Towards the tip, each of these hair has feather-like structures. That just helps the plant trap pollen grains better. Okay, so that's about our corn cob. Example, in the corn cob, the ears, you see the stigma and the style, which wave in wind to trap the pollen grains. Okay. Now let's move on and talk about how water pollination happens. See, just because a plant is aquatic doesn't mean it will undergo pollination by means of water. See, there are so many plants like lotus, water lilies, which bloom in the water. They are aquatic plants. Eichhornia, they are aquatic plants. So flowers there will bloom on the surface. You see how pretty water lily and lotus and Eichhornia flowers are. The same logic that was applied to wind pollination will apply to water pollination also. I told you that if a plant is going to undergo pollination by means of wind, it's most likely not going to be very colorful because wind cannot see, smell, taste, nothing. So it's not going to be modified in those ways. Same will apply for water pollination also. If it is getting pollinated by water, water is also lifeless. Water cannot see, smell, taste. So all of these extra makeup is not required by flowers. But despite that, why is it that lotus flower, lily flower, water lily flower and Eichhornia, they are very pretty colored. Why is that so? That's because they're not pollinated by water. They mostly rely on insects for pollination, or sometimes even wind can pollinate them. The reason is because they are not produced inside water, right? Though they are rooted inside, though the roots are within the water bodies, soil, the flowers are produced at the surface. Since the flowers are produced at the surface, water is not involved in pollination. It's either wind or insect or any other animal. Now, keep that in mind. Just because a plant is aquatic does not mean it is going to undergo pollination by water. Water pollination is extremely rare in angiosperms. There are about 30 genera, that's all, in which you can see water pollination taking place, and among those 30 genera also, most of them are monocotyledonous plants. For a plant to undergo pollination by water, the plant has to be submerged, flowers have to be produced inside the water body. The flowers, when they're produced inside the water body, again it's useless for it to be colorful. So inside the water body, typically, pale colored flowers are produced. You have to learn about how pollination happens by water in two plants. One is Vallisneria, the other one is Zostera. Vallisneria and Hydrilla are examples for freshwater plants that are pollinated by using water. And Zostera is a marine plant. It's called a seagrass that also gets pollinated by water. You have to learn about how pollination occurs in Vallisneria and how it takes place in Zostera. First, let's learn about how it happens in Vallisneria. Vallisneria is a freshwater plant. You have a separate male and a female plant. Male plant produces male flower, female plant produces female flower. Both of these plants are submerged, they are present inside the water body, and the flowers are also produced inside the water body. So this is a female plant that you're looking at here. Okay, so this is a female plant, male plant is not shown here. The female plant produces a flower, one or many flowers, and these flowers have a coiled pedicel. Pedicel is the stalk of the flower. It is coiled spring-like. Once this female flower matures, it will uncoil and reach the surface of the water body. Like it will go and reach the surface, not go beyond the surface, it will reach the surface. At around the same time, if the male flowers have been released or the pollen grains have been released, they again do not have locomotory structures. It's not like they can use flagella and cilia and swim and come and meet the stigma. They will have to rely on water currents. As the water moves passively along with water, they will also move, and if they end up finding the stigma, they will fertilize and form seeds. Once this pollination happens and pollen germination happens and fertilization happens, this coiled uncoiled pedicel will coil back and the flower comes back inside. Inside the flower, this will form fruits and seeds. So if the seeds are formed inside, the seeds will fall here eventually, and then newer plants will arise from here. This is how water pollination happens in Vallisneria. Now this is called epihydrophily. Water pollination is also called hydrophily, and this is called epihydrophily. Hydro means water, philia means love for something, epi means above. Since this is happening above, on top, at the surface level, you call this epihydrophily. As opposed to this, if you look at how pollination happens in Zostera, it's quite different. In the sense, it happens inside the water body and not at the surface. Because it happens deep within the water body, you call it hypohydrophily. Hypo means below. In most Zostera species, they are monoecious. There is one plant, but it produces two different types of flowers, unisexual flowers. Male flowers, female flowers are separate. The male flower produces the pollen grains. The pollen grains here have a sort of unique shape where it is ribbon-like. This ribbon-like pollen grain will swim passively in water along with the water current, and if it comes in contact with the stigma, it will germinate there, bring about fertilization, and eventually seed formation, fruit formation will happen. All of this happens under the water surface. Okay, so here and also in epihydrophily, generally in hydrophily, in order to prevent wetting of the pollen grain, there is a mucilaginous coat present outside it. So it will have a thick mucilaginous covering. In case you're wondering what wetting is, water will have a lot of microbes. Microbes are everywhere. They're in the air, on the land, inside the water body, everywhere. If the pollen grain is not mucilaginous, if it is dry like that, if it comes in contact with water, the microbes in the water can kind of, cause damage in the pollen grain. In order to prevent such things, it has a mucilaginous covering. Mucilaginous covering is like a jelly-like covering. So you can think about it like a Vaseline kind of thick layer present outside the pollen grains, and that will not allow water to enter into the pollen grain. That way it protects the pollen grain for a longer period of time. That's about our seagrass. Now, speaking about animals being used in pollination, though we associate insects and particularly bees a lot with pollination, many other types of animals also have been seen to bring about pollination. For example, you have birds and bats and certain primates, tree-dwelling rodents like mice, reptiles like certain types of lizards. All of these are known to bring about pollination. But among all animals, insects are the most dominant pollinating agents, and among insects is the bees that are the most dominant pollinating agents. Of course, you see wasps and flies and butterflies also bring about pollination, but if you look at the numbers, bees pollinate most of our plants. Okay, so now we'll talk about how a flower needs to be modified if it has to undergo pollination by means of insects. Now, insects are living, breathing organisms, right? They have many sensory structures. They can see, they can smell, they can taste food, they consume food. So, the flowers will have to be attractive enough to grab the attention of these bees if at all they have to be pollinated. Similar to how if you were to impress your crush in school, then what do? Firstly, you will try to look good, right? That is the first appearance. For as long as you look good, that will grab attention. So you wear your nicest clothes and you also want to smell good. So you might spray some perfume or a deodorant and go to college or school. And then these are called attractants. To grab the attention for the first time, attractants will help. What if you're wearing the most, branded clothes and you're smelling very good, but your personality is, it's a waste. Nobody will be interested in you, right? So if they are drawn towards you, there should be some reward that they will get in return so that they keep coming back to you. You should be very good at conversation, or maybe you should be like a very nice person, kind and helpful. Then they will want to come back and talk to you, right? Same thing applies even for bees or insects for that matter. For the first time, for the insects to be drawn towards the flowers, attractants are required. Attractants are required. Attractants. And flowers offer these attractants, flowers have these attractants in the form of being colorful, large, showy, and also in being fragrant. Or let me just write smell here. You know, there are flies that sit on cow dung, flies that like dung, beetles like dung beetles that tend to make balls out of cow dung and excreta, right? Certain insects are found near rotting meat. So it's not necessary that all insects are attracted to good odor. Some insects can be attracted to foul odor also, like some flies are attracted to the smell of cow dung, some flies are attracted to the smell of rotting meat. Flowers have evolved those adaptations too. So here the insects, the flowers may produce either a good fragrance to attract insects like bees and butterflies, or it may produce a foul odor or a bad odor to attract flies and beetles. Either way, it's attracting insects. Here it could be bees and butterflies. Butterfly, butterfly. You remember that? Where are you going? It used to be a viral thing, I think 2 years ago. Yeah. So firstly, these are what the flower uses in order to attract insects. And then you have what is called rewards. Now that the insect got attracted to these flowers by their color or fragrance and have visited the flower, if it gets something in return, then they might visit again. They might consider visiting again, right? So here the rewards include snacks. Basically, it will provide pollen grains to the insect to munch on. some juice in the form of nectar. And in some plants, you will see that the flowers inside the ovary will allow the insect to lay eggs. These are the rewards. Once the insects know that they're getting so much luxury treatment inside the flower, they will not stop visiting the flowers. They will consider that as a cafe. Every time they're tired, they want a little drink, they will go to the flower, they will sip on some nectar, maybe munch on some pollen grains and then leave. In that process, while visiting one flower to another flower, they will end up bringing about pollination. Okay. So here the flowers are large and colorful. In case they are not very large, tiny flowers will cluster together to form what is called an inflorescence. They are also fragrant. Now the color and fragrance is something which is going to attract the flower. These are attractants. Now what are the rewards? Reward is providing nectar, providing pollen grains, and also allowing insects to lay eggs inside them. There are two examples given in your textbooks. One is Amorphophallus and the other one is Yucca. Amorphophallus is one angiosperm plant that produces the largest flower, and this will allow insects to lay eggs inside the flower. Also, you have a plant called the Yucca plant. The Yucca plant is associated with a moth called the Yucca moth. They're both dependent on each other to continue their life cycles. Like the Yucca moth lays its eggs only inside the locule of this flower's ovary. It doesn't lay its eggs anywhere else. Unless it lays its eggs, its generation will not continue. It will not be able to have babies. Likewise, the only insect that can bring about pollination in a yucca flower is this yucca moth. If not for yucca moth, this cannot be pollinated. It is not going to have its seeds, its babies. Without this flower, this cannot make its babies. So they're both dependent on each other for their reproduction and to complete their life cycles. So that's such a cute thing that you can find in the living world, where a plant and an animal are dependent on each other to continue their life cycle, right? So yeah, sometimes the insects won't know. Insects are not given like a task, right? You go to this flower and bring about pollination here. Pollination happens as a byproduct. It's not like the insects are visiting flowers so that they can bring about pollination. It's not like the top thing in their to-do list for every day. It just happens by chance. So sometimes it so happens that the insects visit the flowers, enjoy and sip on some nectar, and end up munching on some pollen grains and, not bring about pollination. So you call that nectar or pollen robbers. Such insects are called nectar or pollen robbers. So with time, plants and flowers also evolve in such a way that they will maximize the chances of pollination. In fact, in some flowers, it is known to have what are called nectar guides. Okay, so our vision is very different from bees and insects' vision. They can see some wavelengths of light which is not visible to us. So apparently on top of petals you have certain guides or streaks sort of things which we cannot see but bees and other insects can see. If the insect goes through that way, it will directly lead it to the nectar, and if it passes through that way, it will almost 100% bring about pollination too. So plants are super clever, much more clever than you and I realize, right? So that's about animal pollination or insect pollination. Yeah, now let's talk about what is called inbreeding depression. I'm sure you would have noticed in your circle or someone you know where they marry very close relatives, right? Sometimes in some cultures in South India, uh, girls marry their mother's brother's son, like their first cousins, mama's child or atta's daughter, they get married. So in a lot of cases, in a lot of such cases, such marriages are called consanguineous marriages. When such marriages happen, the chances of the children having developmental disabilities like mental retardation, not being able to speak and hear, like deaf mutism, genetic disorders, is slightly higher compared to when two entirely unrelated individuals get married and have children. That's because when they are getting married within the same family, they carry similar kind of genes and alleles, and as a result, homozygous genes getting expressed, the chances will be very high. So that is not limited only to humans, that can happen in plants too. In plants, what does it mean when we say closely related individuals get married? It just means self-pollination. repeatedly, generation after generation, if they are undergoing self-pollination, then the offspring that are produced as a result may not be very healthy. They may not grow very well. The yield might be very low. They might not be able to produce seeds and flowers, and their fertility might be reduced. This is called inbreeding depression. Inbreeding means self-pollination. One or two generations of self-pollination will not cause much harm, but repeatedly, if they are undergoing self-pollination generation after generation, eventually you will notice that the plant that is produced is not growing very well. It's not able to, produce flowers. Growth is retarded. All of that can happen. That happens as a result of continuous self-pollination. So plants also know no organism in this world will want to bear children or have offspring that are not good enough. Everybody wants to have the best children in terms of reproductive fitness. You want the species to continue. I told you in the beginning of the chapter that every organism's biological purpose is to make sure that our species continues, right? So here, even these plants, though they are brainless, they know that if at all this happens for a long period of time, if this plant does not produce flowers and not produce seeds, this is not going to have a next generation. Then the entire species is, there's a problem, right? They cannot continue to live. They cannot continue to exist. So plants have clever, super intelligent mechanisms and devices to kind of ensure or maximize the chances of cross-pollination and kind of reduce the likelihood of self-pollination. Whatever devices plants have evolved to encourage cross-pollination and discourage self-pollination are called outbreeding devices. Outbreeding is cross-pollination. Inbreeding is self-pollination. Inbreeding is not good for the plant, for the species. Outbreeding is great because variation. Okay. So there are 4 such outbreeding devices about which you will have to learn. It's very easy, common sense. If you use, you will be able to remember it for a long period of time. This again is like a love story only, like between one hero and heroine, there are numerous reasons why there will be rifts, right? So it's possible, like the first reason for, the first outbreeding device that is mentioned in your textbook is that whenever in bisexual flowers, that is where self-pollination can happen, like autogamy can happen, whenever pollen grain is mature and about to release pollen grains, the stigma is not yet mature, or the stigma becomes receptive first. At that time, pollen is not yet mature. So there is age difference. You can think about it like that. In one family, 2 cousins, they want to get married. They cannot get married. One of the reasons is because the girl is right now 7 years old and the boy is 27 years old. Or the boy is 7 years old and the girl is 27 years old. So it's impossible, right? So like that, here also, whenever over here, both anther becoming mature and stigma becoming receptive will not happen at the same time. There is no synchrony here. So pollen release and stigma receptivity are not synchronized, which means it will not happen at the same time. So here, first the pollen is released, the stigma won't be receptive at that point. Even if the pollen falls on the stigma, stigma won't be able to do much. Pollen germination will not happen. It's a waste. If the stigma is mature first and the pollen is not yet released, then it's good, right? The same pollen will not go and fall there. So that way, it kind of encourages cross-pollination. Secondly, there is a difference in position. Over here, it is physical position, but you can think about it for memory, financial position, right? So some people are very poor and some people are like very rich, and there is a status issue there when 2 people fall in love. So that is one of the reasons why they might not want to get the 2 married. Like one cousin is from a lower economic background and another one is like super rich. When that happens also, that marriage may not happen, right? So likewise here, the anther and the stigma's positions are so different and distant within a flower that even if the pollen grains get released, the chances of it going and landing on the stigma of the same flower are very, very minimal. In all of these devices, you can't ensure like 100% of the time that it is going to undergo cross-pollination itself. Sometimes, very rarely in such conditions, self-pollination also could happen. But mostly it will try to encourage cross-pollination and prevent self-pollination. The third device is almost fail-proof. So here, this is a genetic mechanism. It's called self-incompatibility. If if the pollen grains that land on the stigma have the same genes as the stigma or as the plant which is bearing the female flower, then it won't even allow it to form the pollen tube. I'll give you an example. Here you have the ovary, here you have the stigma, here you have the pollen grains. Okay. So the stigma here has two genes, S1 and S2. If a pollen grain carries S1 gene or S2 gene, pollen germination will be prevented. Outrightly, the stigma will say, no, you're like my brother, I cannot do this, I never saw you in that way, and reject it. If at all it carries any other genes like S3, S4, anything but S1 and S2, then it will get a green signal, it will be allowed to form the pollen tube. This is one way to ensure that cross-pollination itself happens. This is called self-incompatibility. For as long as it's from the same plant, it's going to be incompatible. And this is a genetic mechanism, which means it is determined by the kind of genes that are present in the stigma and the pollen grain. Finally, we have production of unisexual flowers. There are two cases here. The plant may be monoecious or it may be dioecious. If it is monoecious, as in the case of maize and castor, in maize we just now saw you have same plant, you have the male flowers being produced at the apex. These are called the tassels, the male inflorescence, right? The male flowers are produced here and the female flower is produced here. Okay, so the female flowers are going to be here. Since male flowers don't have pistil, female flowers don't have anther, autogamy is impossible here. I told you for autogamy to take place, the flower 100% has to be bisexual. In this case, autogamy does not happen. There is no chance for autogamy. But gatonogamy, within the same plant, pollen transfer can still happen. Gatonogamy, there is a chance. Coming to dioecious plants like papaya, here you have a separate male plant and a separate female plant. Male plant produces male flowers having stamen. Female plant produces female flower having pistil. So 100% xenogamy only can take place here. So over here, both autogamy as well as gatonogamy can be prevented. Okay? So that is about our 4 outbreeding devices. Now we have the pollen-pistil interaction. This is so interesting. This is like a masterclass in consent. You know how a lot of people don't understand that a no means no. They will, force you to do things you do not like. But pollen-pistil interaction in plants, it's like they understand even though they are brainless. Plants understand that a no means no better than most humans do. So I'll tell you how this works, okay? So here you have this pistil, right? In the pistil, you have a stigma, and then you have the style, and then you have the ovary. To the ovary, within the ovary, there's this placenta, and to the placenta, you will find ovules attached. Now, inside the ovule is where the embryo sac is present, and within the embryo sac, you have the female gamete. Now, the pollen grain that is released by the anther Oh my god, my pollen grain and anther look very ugly. So let's do this. Okay, so the pollen grain is released by the anther. The pollen grain is carrying two male gametes inside, and this via the germ pore will have to form a pollen tube. The vegetative cell forms the pollen tube. Pollen tube enters the ovule, and inside the pollen tube, the two male gametes that are there, that will also gain entry, and ultimately fertilization is brought about. I told you how this stigma here acts as the landing platform earlier. Stigma is the landing platform. This is like a queen, okay? So this has so much power. It can accept or reject pollen grains. Now, if you look at nature, it's not like all the rose plants are in one corner of the world, all of the jasmine plants are in another corner of the world, right? In nature, you will see many different species of plants growing right side by side. So, it's possible that the pollen grain of rose ends up landing on the stigma of hibiscus plant, and the pollen of hibiscus plant can end up landing on the stigma of a mango flower. So, this naturally happens in nature, right? Naturally happens in nature. Okay, whatever. Yeah. So here, it's possible that pollen grains belonging to different species can end up landing on the stigma. If this is a pollen grain from a different species, it can't bring about fertilization. See, even if you look at animals in the ocean, there are so many different species of fishes. In fishes, you see external fertilization. So, all of the sperms and the eggs are going to be released out in the open in water. So, you don't see that a shark's egg fertilizes a clownfish's egg, or some other fish fertilizes another species of fish's egg, right? That doesn't happen. Same way here also it does not happen. Now, there has to be some way that the stigma recognizes which pollen belongs to the same species and which one doesn't, right? So, how does that happen? That happens by this genius mechanism where chemicals talk to each other. On the surface of the pollen grains, there's going to be some chemicals. Okay? So, there are chemicals present on the, what size is this? Okay, so, there are chemicals present on the surface of pollen grains. Also, there are chemicals present on the surface of stigma. So, the chemicals that are present on the surface of stigma interact with the chemicals that are present on the surface of pollen grains, and depending upon whether they belong to the same species or not, the stigma can outrightly reject it, like swipe left and say, no, we are not good, we are not compatible, this is not okay, you belong to an entirely different species. It's like a giraffe is trying to mate with a mouse. It is not going to work like that. So, the stigma can reject it for as long as it belongs to a different species. What if it's the same species? Then there are two scenarios. Either this belongs to the same species. Oh my god, the spelling is wrong. Just give me a second. These lights are blinding me. Same species, different plant, or it can be same species, same plant. If it is the same species and same plant, in plants that have self-incompatibility. Self-incompatibility is something that you don't see in all plants. Some plants have self-incompatibility where if the pollen belonging to the same plant lands on it, it will not allow it to form a pollen tube. I told you about that in outbreeding devices. If it is a plant species that has self-incompatibility, then this also is going to be rejected. Now, when this pollen grain approaches the stigma and says, you're kind of cute, I kind of like you, maybe we should be more than friends, the stigma is like, are you out of your minds? Are you serious? What do you mean? We both belong to the same plant. We're like born to the same parents. We're like siblings. We're like brother and sister. We can never do that, and it will outrightly reject this pollen, brozoning it, but that does not apply to all plants, I repeat. In plants where incompatibility does not exist, this can still occur. Okay, so that depends on the plant species. This may or may not happen. Okay, so in case there is self-incompatibility, understand that this is what that means, incompatibility, then it will be rejected. But if it is the same species but a different plant, it's like a win. Same species, different plant, even if it's even if it has self-incompatibility, even then it will accept that particular proposal and they will get married and live happily ever after. So what do I mean when I say it's going to reject some pollen and accept some pollen? When I say it rejects some pollen, I mean to say that it will not allow that pollen grain to form a pollen tube at all. It just sits there and just rots away. But if it accepts it, only then will it allow the pollen tube to form. Okay. So the other pollen here, just because the pistil rejected it by saying that we are not cut out, we are from different species, or we're like brother and sister, whatever be the reason, when the stigma says it's a no, the pollen respectfully agree to it. They do not force the stigma and force penetrate themselves into the style. And they won't even go on to blame the stigma and saying, if you did not want it, then why are your petals so colorful, right? So they don't. That doesn't happen here. So a no is very well respected. These pollen grains have more maturity than most humans you can find on our planet. That's what I meant when I say this is a masterclass in consent, and pollen grains understand that a no means no better than most humans. Okay. So that's about pollen-pistil interaction. You can see that just because a pollen has landed on the stigma does not mean fertilization and pollen tube formation will happen. It depends on compatibility. For as long as the pollen grain is compatible, pollen tube germination will be allowed. So the pollen accepts it, otherwise it rejects it. How will a pistil get to know whether it is compatible or not? By chemical interaction. So there's a chemical dialogue that takes place. They communicate not by words but by means of chemical interaction. The surface of the pollen has chemicals that interact with the chemicals that are present on the surface of the stigma, and that is how it realizes whether it's a swipe left or swipe right. Okay. Yeah, that's that. You can see over here how some of the pollen grains are allowed to form a tube while some others aren't. Here is a real micrograph of a stigma. You can see all of these tiny dots on the surface are the pollen grains. They're trying to penetrate the stigma. A lot of these pollen grains will penetrate the stigma, and depending upon how many ovules are present inside the ovary, fertilization will happen. Not every pollen grain can bring about fertilization. A lot of them do bring about fertilization, similar to how many sperms are released after ejaculation, thousands of them, but there's only 1 or 2 eggs per cycle. Only some of them will be successful in fertilization, right? Yeah. Now, all of the events from the time the pollen grain is released from the anther and deposited on the stigma to the time the pollen tube forms and enters the ovule, every event that takes place is called pollen-pistil interaction. Okay. All the events from the pollen deposition on the stigma till the time pollen tubes enter the ovule are called pollen-pistil interaction. Now the pollen tube has formed here, it passes through the style, it has entered the ovule, it enters the ovule through the micropyle. I told you the reason the micropyle exists is to enable the entry of pollen tube. The pollen tube is not coming, alone there. The pollen tube within itself is carrying the 2 male gametes. The pollen tube is formed from the vegetative cell. Keep that in mind. So, when the pollen tube enters through the micropyle, towards the micropyle, which are the cells that are present in the embryo sac? It is the egg apparatus. You have 2 synergids on either sides, and at the center you have the egg cell. So, ultimately the male gamete is supposed to fuse with the egg cell, but it does not directly penetrate the egg cell. It will penetrate one of the 2 synergids that are present on either sides of the egg. Basically, the filiform apparatus that is present at the base of the synergids, that is what will guide the entry of the pollen tube. So, this filiform apparatus this one guides pollen tube entry. So, once it enters into the synergids, see what it does? It releases both of the male gametes into the embryo sac. Next, fertilization will happen. So once the pollen tube releases the two male gametes inside, one of the male gametes fuses with the egg. This is one male gamete. This fuses with the egg, which is the female gamete, and this ends up becoming zygote. The male gamete is haploid, female gamete is haploid, the zygote becomes diploid. What happens to the other male gamete? If you look at the structure of the embryo sac, where are you now? Oh my. Yeah. So if you look at the structure of the embryo sac here, look, here is where the pollen tube is entering. Here is where the two male gametes are released. One male gamete fuses with the egg cell here. The other male gamete fuses with these two polar nuclei. Okay? So I will erase this so you don't get confused. How far away is this? Yeah, found it. So the other male gamete fuses with the polar nuclei. N plus 1, N plus N. Sometimes this fuses to form what is called the secondary nucleus, which is diploid. And they end up forming a triploid structure called the primary endosperm nucleus. Since in angiosperms, two fusion events are taking place. This male gamete is fusing with the egg cell to form the zygote. The other male gamete is fusing with the polar nuclei to form the endosperm. Because two fusion events are taking place, you call this double fertilization. Up until pollen-pistil interaction, we've learnt about pre-fertilization structures and events. Now we're moving on to double fertilization. There are two fusion events taking place. The first one is syngamy, second one is triple fusion. Syngamy is the normal term that you use for fertilization where a male gamete is fusing with the female gamete. So it forms the zygote. Triple fusion, because three nuclei are involved here, two polar nuclei that are haploid and a male gamete which is also haploid. This ends up forming a triploid nucleus which is called primary endosperm nucleus, or PEN in short. Now, once the male gametes are released inside the embryo sac, the formation of zygote, the formation of primary endosperm nucleus, both happen simultaneously. Eventually, what's going to happen? The zygote is the first cell of the next generation, right? The zygote is the first cell of the baby. Eventually, when you sow the seed, a little plant is going to come outside. That is going to come out of a multicellular structure called an embryo. The zygote is one cell. Embryo has many cells. This zygote will undergo mitosis to give rise to a multicellular embryo. I want you to understand where all of this is happening. So we have the ovule, right? It is within the ovule that the embryo sac is there. It is within the embryo sac that the embryo and endosperm are forming. Eventually, this ovule is going to become the seed. So it is inside the seed that the embryo and the endosperm are forming. This embryo portion present inside the seed is what is going to give rise to the future plant. When you sow a seed, a new plant sprouts and arises from it, that is going to form from the embryo. Okay. So, this development of zygote to embryo happens within the ovule itself by undergoing mitosis and differentiation. What about primary endosperm nucleus? That is the name of the triploid nucleus. The cell is called primary endosperm cell. This primary endosperm cell will undergo mitosis. This is also a single cell. This will undergo mitosis and give rise to a multicellular endosperm. What is the function of this endosperm? Embryo is going to give rise to the baby plant. What is the endosperm going to do? Endosperm has a function of nourishing the developing embryo. If the zygote has to undergo cell division and give rise to embryo, embryo has to differentiate, it requires nutrients. And the nourishment that is required for the development of zygote to the embryo is provided by the endosperm. So this is a nutritive tissue that is triploid in nature. Though zygote formation, primary endosperm cell formation happens at the same time, endosperm development happens faster compared to the embryo development. Okay, so this happens faster compared to this. This is relatively slow. This is same time. This fusion, same time. From this to this happens faster compared to from zygote to embryo development. I'll tell you the reason for it. For zygote to develop into embryo, nourishment is required. That nourishment is provided by this. Unless this food is available for the zygote, it will not be able to develop into the embryo. For this to happen, this is a prerequisite, this is a requirement. Without energy, this growth and development cannot happen. In order to ensure that the zygote develops into the embryo, this development of endosperm has to happen first. Okay, so in your textbooks it's given that the development of the development of endosperm precedes the development of embryo. Precedes means it happens sooner than embryo development, because this is what is providing the required nourishment for the development of the embryo. So this forms first. Okay. Syngamy triple fusion. These are the two fusion events that happen during double fertilization. So first endosperm develops, then zygote develops into the embryo. First we learn about how endosperm formation happens, then we learn about how embryo formation happens. We'll begin with endosperm development. Endosperm, what happened to this? Wait, this is not working. Why won't you work? Never mind. So we had the primary endosperm cell, which is triploid. Okay. The central cell itself will transform into the primary endosperm cell after triple fusion. This has a triploid nucleus. If you remember, we spoke about how during female gametophyte development or embryo sac development, numerous mitotic divisions will happen. 3 mitotic divisions will happen. Initially, karyokinesis happens, immediately not followed by cytokinesis. So it leads to a multinucleate condition, right? I gave you the same, I told you the same about tapetal cells also. The reason they are multinucleate is because karyokinesis happens and that's not immediately followed by cytokinesis. Similar thing happens here also. This primary endosperm cell will undergo numerous mitotic divisions where karyokinesis happens, cytokinesis does not immediately happen. As a result, you end up getting a structure that is multinucleate. Every nucleus here is going to be triploid. So this is called a free nuclear endosperm. Eventually, much later, cytokinesis takes place and cell walls are laid between the nuclei and it forms what is called a cellular endosperm. Okay. Now, one example for the endosperm that we consume is coconut. The tender coconut water that we consume, that is free nuclear endosperm. Okay, so here you have the tender coconut water. This is example for prenuclear endosperm. Let me write it here. As this prenuclear endosperm develops into cellular endosperm by producing cell walls between the nuclei, you get, if you leave the coconut as it is for a few days, you will notice that the water will harden to form the kernel, coconut kernel, that white part that we use to make kopri mithai. Right. So this is our coconut fiber broke open. This coconut kernel is the example for cellular endosperm. Okay. In dicots and monocots, in both cases, this is how the initial development of endosperm will take place. Next, we have to learn about, wait, there's some notes here. Triploid tissue endosperm, it is filled with, there's too much yellow, let's use blue. Reserve food, it is nutritive, it is meant to be used for the developing embryo. Initially, many nuclear divisions will happen, which will give rise to free nuclear endosperm. Later, cell wall formation will happen, that will give rise to cellular endosperm. The tender coconut water is free nuclear endosperm, and the white kernel of coconut is the cellular endosperm. Okay, so this endosperm that is present inside the seeds may completely be consumed during embryo development, or some of it may still be retained within the mature seed. See here, within the same seed, you have embryo, you have endosperm. Endosperm is the food for the embryo. As the embryo is developing, endosperm will start getting reduced in quantity, it will start getting used up. So, in a mature seed, endosperm may or may not be present. It may or may not be retained till maturity. So, there are some seeds in some species where endosperm is completely used up. For example, in dicot plants like peas, beans, groundnut, you will not see any endosperm left in the mature seed. As opposed to that, in plants like coconut and castor, you will see endosperm remaining even at maturity. Okay. Now let's talk about the development of embryo. Embryo develops from the zygote, right? The zygote is the first diploid cell. This will undergo mitosis. Once mitosis happens, the cell divides to give rise to two cells. Eventually, that continues to divide to give rise to more cells. And the first few stages of the embryo development I'm showing you here. The initial stages of development of embryo again are going to be pretty similar to monocots and dicots. The later developmental stages vary, but that much detail you do not have in your syllabus. So for now, one of the first few phases that you see is the globular embryo. Over here you find a spherical sort of structure, and you have like a long filament of sorts present. So this is called the embryo proper. This is called the suspensor. This embryo proper is going to give rise to the future plant. What is the function of the suspensor? The suspensor penetrates itself into the endosperm tissue. This is like the endosperm. Imagine it in 3D. Okay. So inside a seed, you have the embryo on one end, you have the endosperm on one end. Embryo is supposed to derive the nourishment from the endosperm. So this suspensor penetrates into the endosperm, draws nourishment from it and passes it on to the embryo proper. So this can develop into the future plant. Okay, so this is the suspensor. This is not directly involved in formation of future plant. This helps absorb the nourishment from the endosperm and pass it on to the embryo proper. Eventually, later, this globular, spherical thing will become like a heart-shaped embryo. The suspensor will still remain. Later, it will develop into the mature embryo. So the correct sequence would be globular embryo to heart-shaped embryo to mature embryo. We have to learn about the structure of dicot embryo as well as monocot embryo. In an embryo, you will have what is called an embryonal axis. This is our embryonal axis. Do you ever feel? At the center of, not at the center, somewhere on the embryonal axis, you will have what is called a cotyledonary node. Similar to how you have nodes on stems, even in the embryonal axis, there will be a node to which, in this case, since it's a dicot embryo, two cotyledons will be attached. These two are the cotyledons. This portion of the axis above this point of attachment is called the epicotyle. Epi means above, cotyle, point of attachment of cotyledons. This is epicotyle. This bottom portion is called the hypocotyle. Hypo means below, cotyle, cotyledon. At the tip of the epicotyle, you have the plumule. The plumule in future is going to give rise to the shoot system. Here, the tip of the hypocotyle is called the radicle. The radicle is going to give rise to the future root system. I've drawn this embryonal axis very long, but in reality, if this is like a dicot seed, the embryonal axis is going to be just very, very small like that. But I can't show you all the parts if I make it so small, which is why I drew it long. It's not going to be so long, okay? So it's going to be short. In this diagram, I'll show you where the embryonal axis is. This is our embryonal axis. This is the embryo proper. Here you have the suspensor. To this point, the two cotyledons are attached. At this point, here you have one cotyledon that I'm highlighting or outlining in orange. And here you have the second cotyledon. This portion above the point of attachment of cotyledon is the epicotyle. The tip of that is your plumule. This portion below the point of attachment is the hypocotyle. The tip of the hypocotyle is your radicle. On top of the radicle, you have what is called the root cap. So that is the structure of a dicot embryo. A monocot embryo is also pretty similar. Here also you have an embryonal axis. Since this is monocot, you have only one cotyledon. That single cotyledon itself is very large and shield shaped. We call that scutellum. Instead of another cotyledon, there is a small extension of sorts that you can find, which is called epiblast. Point of attachment is the cotyledonary node. Above you have epicotyle. Below you have hypocotyle. Tip of the epicotyle is the, let me just write it like, tip of the epicotyle is the plumule. This gives rise to the future shoot system. Tip of the hypocotyle is the radicle. That gives rise to the future root system. Apart from that, you can find a sheath-like structure covering the plumule, which is called coleoptile. Right on top of the radicle or the root cap, you can find a sheath-like covering, which is called the coleorhiza. So, terms like scutellum, coleorhiza, coleoptile are exclusive for monocot embryo. You don't have those terms in dicot embryos. Coleoptile and coleorhiza doesn't exist in dicot embryo. If you look at this structure, over here you have the embryonal axis. Here you have the single large shield-shaped cotyledon called scutellum. At the tip of the epicotyle, you have the shoot apex or the plumule, and then you have this coleoptile. At the radical tip, you have the root cap, and surrounding the root cap, you will have the coleorhiza. This small projection of sorts is the epiblast, that is the reduced, highly reduced cotyledon. That is the structure of monocot embryo. Again, the axis may not be this long. I'm only showing it so long. So because I want you to understand what are the different parts present in it. Now, we'll move on and learn about artificial hybridization. See, when you go to buy something, some clothes for yourself, you find a top that is such a beautiful color. It's like a nice bright pink or a purple color. Okay, whatever color you like. Nice color that you like, you're fond of. And then it does not fit you properly. But then you find another outfit that is like perfect fit, but it is in a color that you do not like. You wish to have a good fit in the color that you want, right? So you will ask people in the shop to find you a fit that is good in a color that you like. Plant breeders do this all the time with respect to plants. Like you know how many species of plants that are commercially cultivated have many varieties. When it comes to hibiscus plants, there are so many varieties. You see hibiscus flowers that are red in color, some of them are orange, some of them are pink. And even rose flowers come in so many different colors. Mango, there are so many varieties. So within the same species, many varieties will be present. Sometimes it so happens that the farmer or the plant breeder requires two traits. These two traits are present in two different plants. You want to combine them in one hybrid. What do you do then? You perform artificial hybridization. You try and ensure to make sure you cross the two plants, like one plant has very pretty flowers that you want, another plant has disease resistance that you want. You want to combine this pretty flower with disease resistance into one hybrid. You yourself artificially bring about pollination between these two plants. That is called artificial hybridization. How exactly that is performed, I will tell you. So let's say there are two plants, two varieties belonging to the same species. Let's consider. So this is variety 1, variety 2. Let's say the first variety produces some not so commercially flowers that are commercially not so much in demand. It produces pale colored flowers that are small in size. Okay. So let's say this produces small flowers that are not in demand, but it has pest resistance property, which is a good thing. And the second variety produces very large showy flowers that are in demand but does not have pest resistance. This produces large flowers. Even for flowers we are doing body shaming, see. Large flowers in demand, no resistance against pests. So what does the plant breeder want now? The plant breeder wants this pest resistant property from variety 1 and large flowers from variety 2, and they want to combine both these traits in one hybrid variety of plant. This is their idea. But sometimes, unfortunately, it can so happen that the hybrid plant can end up getting small flowers with no resistance also. To ensure that you 100% get the traits that you desire, biotechnology is the best way. Okay, so we learn about that in biotech chapter. So if at all the plant breeder wants to bring these two traits in a hybrid, what are some of the steps that he will need to perform? He or she will understand. So let's take this flower, assuming they are both bisexual for now. Let's take the two plants. We don't need plants, we can just draw flowers. You have one flower, and you have this other flower. Let's assume they're both bisexual. This will have its stamen. This will also have stamen. This will have the pistil. This will also have the pistil. So they're both bisexual. But here, before you perform the experiment, you have to designate one plant as female and one plant as male. For convenience, I will assign this as female and this is male. Either of them can be male or female. If this one is female, I am going to dust the pollen of this one onto the stigma of this. What I want is to dust the pollen from this flower onto the stigma of this. I do not want the pollen from this flower ending up on this. This is something that I absolutely do not want. Also, I do not want some random flowers, pollen grain of the same species. I do not want some random flowers, pollen grain also ending up on this stigma. So I have to ensure two things right now. I want to make sure that this pollen itself lands on this. I also want to make sure that self-pollination does not happen. I also want to make sure that any pollen other than this does not land on the stigma. Okay. So what does the plant breeder do? Firstly, in order to stop this from happening, the plant breeder will perform what is called emasculation. What is emasculation? Masculine means what? Related to maleness. Emasculation means removing that male part. If this anther matures and releases pollen grains, only then will it go and get deposited here, right? What are we going to do? Even before the anther matures, you will cut these away. Okay, you cut these anthers using forceps even before it matures so that you prevent self-pollination. So why is emasculation done? In order to prevent self-pollination. Now, how do you prevent this pollen from depositing on this? You can do that by tying a bag around this flower, which is called bagging. You tie a polythene bag around it to make sure that no other pollen will get deposited on its stigma. Once this stigma becomes receptive, this anther matures, you take this anther, dust the pollen on top of the stigma and tie a bag again. That is called rebagging. That will ensure that only the desired pollen lands on the stigma and brings about fertilization. So once you tie the bag after dusting this pollen, you allow it to be the same. You will not disturb it. Eventually fruits and fruits and seeds will form within the bag itself. Once the fruits are formed, you will open the bag, you will take the seeds and sow them and you will get the next generation. In the next generation, because this is sexual reproduction, different combinations of these traits you would have gotten in the offspring. Whichever one has the combination that you want, you will select. Okay. So that is what needs to be done. Now, in this example, I took both the plants that were bisexual. They had both male and female parts. If the plant species that we're looking at is like a maize plant, it's unisexual, let's say. If the flowers are unisexual, then what do? We don't need to perform emasculation because the flower in itself is going to have only androecium or gynoecium. It will not have both. So when you're calling a plant or a flower the female parent, it has only gynoecium. There's nothing over there extra to cut away. You don't have to prevent self-pollination because it's not going to happen there, right? So you can avoid the emasculation step for as long as the flower is unisexual. So first step is emasculation, that is done to prevent self-pollination. Second step is bagging to prevent contamination from unwanted, undesired pollen grains. Then you dust the pollen, then you bag it again. Fruits and seeds will develop within the bag itself. Okay? Yeah. So now again, after fertilization, we started talking about the post-fertilization structures and events, embryo, endosperm, all of that will come in post-fertilization structures. Also seed and fruit. As the zygote develops into the embryo, primary endosperm cell develops into the endosperm. The seed will develop into the ovule and the ovule will develop into the seed and the ovary will develop into the fruit. Now we're talking about all of these are events that happen post fertilization. So far we spoke about how embryo development happens, how endosperm development happens. Now we have to talk about how does the seed form, how is it different from the ovule, and what is its function and all of that. Okay. So seed is the final product of sexual reproduction. In most cases, seeds form only after sexual reproduction, after fertilization has taken place. Remember, I'm saying most cases and not all at all times. So at most times, only after fertilization has happened does seed formation happen. Since seed is formed after fertilization of ovule, you can call the seed as the fertilized ovule too. Within this fruit is where the seeds are formed. So they are formed inside the fruits. What are the different parts that you can find in a seed? In an ovule, you had two integuments that were protective. In the seed, you will have seed coats instead of the integument. Integuments will transform into two seed coats. Within that, you will have the embryo. Obviously, the embryo will have its own axis, and to the axis, the cotyledons are attached. And in some seeds, endosperm will be present, in some it is absent. Next, we have to learn about the two different types of seeds based on the presence of endosperm. Endosperm at maturity. In all seeds, endosperm will definitely form because in all angiosperms, double fertilization takes place, triple fusion happens. Endosperm will most certainly form. But the question is whether or not it is retained in the mature seed. I told you that the endosperm's function is to nourish the development of the embryo. When the embryo is developing, embryo is growing bigger, the endosperm tends to become smaller and smaller in quantity because it's getting used up. In many species, the endosperm is completely consumed by the time the seed matures. So, in the mature seed, endosperm will not exist. However, in a few seeds, you will find that the endosperm exists even at maturity. Now, depending upon at maturity, if the endosperm is present or absent, you can classify seeds into two types. The first one is called non-albuminous or non-endospermic seeds, and the other one is called albuminous seeds. In non-albuminous, it's self-explanatory, as the name itself indicates. The endosperm residue will not be present. Examples for that includes peas and groundnut. And albuminous seed, example, you have wheat, maize, barley, castor, all of these. Okay, so in this, a part of the endosperm is retained even at maturity. Next, there is something called perisperm that we have to talk about. Peri means around. Peri means periyamma, Allah, around. Sperma means seed. See, this is our ovule. This is the outer integument. Then you have, ouch, this was a very bad line. This is the outer integument. And then you have the inner integument. Inside that, we had the nucellus, right? And within the nucellus, the embryo sac formed. Within the embryo sac, the endosperm and embryo formed, right? So here you have endosperm as well as embryo. In most cases, what happens is, as the embryo and endosperm start to develop, the nucellus will start degenerating. Okay, so nucellus also will start getting used up, it will start getting degenerated. And in a mature seed, this nucellus will not exist. However, in some species like beet and black pepper, you can see that even in mature seeds, some amount of nucellus exists, and that is called perisperm because it is present around the sperm, right? Around the seed, towards the periphery, right? So this residual persistent nucellus is what is referred to as the perisperm. It's present peripherally within the seed. The nature of the perisperm or the ploidy of the perisperm is going to be diploid. Endosperm is triploid, perisperm is diploid. Do not get confused. Now, here is an NCERT diagram. You have a few seeds that are given. The different parts of the seeds are labeled. So, you can see how The different parts are shown here for your reference. Next, what is the advantage of seeds to angiosperm? Seeds offer so many advantages. See, if you look at plant kingdom, not all plants produce seeds, right? Only the gymnosperms and angiosperms produce seeds. The seeds of the seeds of angiosperms are covered by fruit wall, which is even more better and advantageous compared to the gymnosperm seed. It is within the seed that the embryo is present, and it is through the seed that the next generation of plants is obtained by sexual reproduction. So, seeds, these flowering plants have evolved many mechanisms for seed dispersal, especially these fruits. Okay, you have different types of fruits. You have fleshy fruits and dry fruits. Dry fruit does not mean your cashew and pistachio and all of that. Dry fruits botanically is any fruit that does not have a fleshy wall. Okay, botanically, cucumber is a fleshy fruit, apple is a fleshy fruit, strawberries are fleshy fruits, guava, mango, all of these are fleshy fruits. And peas, beans, groundnut, all of these are dry fruits because the fruit wall is not fleshy. Okay. If a fruit is a fleshy fruit, then I'll tell you how the seeds are dispersed. Seeds are dispersed means from where it is produced to going to some different areas and germinating there. It's dispersed to far off areas. In fruit producing plants, what happens is, fruits are food for many animals. You see many parrots and different birds feeding on fruits. You see many animals like squirrels also feeding on fruits. So these animals will not stay on one tree at all times. Let's say there is one tomato plant and some animal comes and feeds on that tomatoes. And in the process of consuming the flesh of the fruit, they will end up feeding on the seeds as well. So they leave that place, go to some other place, and they defecate in some other place. As it is, it would have been impossible for the plant to take the seed, walk and sow it somewhere else, because plants do not move, right? However, because of the production of fruits in angiosperms and animals consuming these fruits, through the once the seeds are consumed by the animal, the seeds will go through the digestive tract and eventually be excreted along with its excretory waste, right, or digestive waste. So wherever it goes and defecates, over there some plant seed will be dispersed. That is beneficial for that particular plant, because if at all there is no mechanism for dispersing seeds to far off habitats, a mango tree produces so many mangoes in one season, and all of those mangoes fall right down there and all of them germinate right there. It will be competition for the parent plant itself, because the soil has limited nutrients. All of those seeds cannot germinate and grow right there. It cannot be supported by the limited nutrients in the soil. So it's best for the plants if the seeds are dispersed to far off locations, so that the geographical distribution of that species also, kind of expands. It's beneficial in another way, because let's say different habitats have different weather conditions. In some place the weather might not be optimal. In some places the weather might be like just perfect. So if all the plants are present of a particular species are present in one area itself, then if the weather conditions go bad there, then they could possibly all die. If they are distributed in different areas, then one or the other will survive and continue the species, right? So not only do fleshy fruits have mechanisms for dispersal of seeds, even dry fruits have. If you leave lady's finger on the plant itself without plucking it out, eventually after the fruit matures, the fruit wall itself will break open and release the seeds. So in one or the other way, these fruit producing plants will have ways to disperse the seeds to far off habitats. And also, these seeds have sufficient food reserves. If the fruit falls off on the ground also, let's say there was no animal that carried it to a distant place, let's say the fruit fell off right there. In the fruit there is organic matter that can be used as nourishment, and also within the seed, either in the form of endosperm or in the form of cotyledons, there will be food reserve that can be used up by the plant until the seed germinates, produces leaves and performs photosynthesis. Up until then, it can be supported. Its growth and development can be supported by the food reserves. Also, talking about protection of the seed, it has a very hard seed coat that offers protection to the young embryo present inside from the harsh environmental conditions or the surrounding environmental conditions. Then, Because seeds are a product of sexual reproduction, meiosis is taking place. During meiosis, crossing over happens. Because of that, the seeds will have newer genetic combinations, and that will increase the variation of that particular species in nature. And seeds are the basis of our agriculture. As the ovules transform into seeds, they will lose water and get dehydrated. They will just retain enough water for it to stay alive. And for the bare minimum metabolic reactions, whatever water is required, that's all it retains, and it loses most of the water. Now, this is important because wherever water is there, moisture is there, microbial infestation can happen. If inside the seed microbes somehow gain entry and there is water there, they will take over the entire space, right? Micropyle is a small pore, but that is sufficient, more than sufficient for microbes to gain entry. But if it is, dehydrated, then microbes cannot survive there very well. By being dehydrated, it's protecting itself against microbial infestation. And another important property that our seeds have is dormancy. Dormancy is staying inactive. That means seeds immediately after they are formed, they do not germinate. Look at the grains we use in our houses and the pulses. All of these are seeds, right? You have wheat, you have rice, you have ragi, and you have green gram. Imagine all of them, germinating and producing plants the day you got them home. You wouldn't be able to store them. You wouldn't be able to have food throughout the year. You can't store any grains if it germinated as soon as they were produced. Them, the seeds being inactive is what is called dormancy. Now, why are seeds inactive? That's because, not because we can store our food, that is because unless the seed gets a signal that the conditions around it is favorable, it's not going to germinate. You store the green gram in a dry, airtight container, it's not going to germinate for many months. But the moment you soak them in water, just after a few hours, you will see that it germinates. It requires that presence of moisture and a little bit of darkness maybe in order to germinate. Unless it gets that signal, it will not germinate. Because of this property of dormancy, you can store this for a long period of time and use it as a source of food for our everyday meals, and also farmers can store it for their next season to raise crops. Okay, so these are some of the advantages that we have for seeds. Speaking of seed viability, similar to how pollen grains have a viability period, seeds also have a viability period. For pollen grains, it was the period during which it retains the ability to form a pollen tube. For seeds, it is the period during which it retains the ability to germinate. Okay, so for different species, the pollen viability varies. For some, it is less, just a few months. For some, it can be many years. There are two examples that are given in your NCERT textbook. One is of Lupinus articus. This is a plant, this is a seed that was obtained during excavation of the Arctic tundra. Polar region where there's a lot of ice for the most part of the year. So, within the ice, they excavated a seed that was apparently 10,000 years old, not 10 years, 10,000 years old. Even after obtaining it, after 10,000 years, for 10,000 years, apparently it was dormant, and once it got the right conditions, it not only germinated, it also produced flowers. So that is how long the seeds can remain dormant. Another example is Phoenix dactylifera, which is our date palm. This was during the excavation of King Herod's palace that is near the Dead Sea. They found a seed that was 2,000 years old, which also was viable and germinated after being provided with the right kind of conditions. Now next, moving on to fruits. After fertilization, as the ovules transform into the seeds, the ovary will transform into the fruit. The fruit wall is called the pericarp. Within the fruit wall, you can find the seeds being present. Now, there are many different types of classification of fruits, one of which is based on the nature of the pericarp, the outer wall of the fruit, whether it is fleshy or dry. If it is fleshy, you call it a fleshy fruit. If it's dry, you obviously call it a dry fruit. Examples of fleshy fruit include guava, orange, and mango. And examples of dry fruit includes groundnut, mustard, etc. Like I told you, groundnut you might not consider as a fruit, but botanically that develops from the ovary itself, and that is called a fruit. So the wall of the fruit is not fleshy, that's why we call it a dry fruit. Now there's another classification of fruits based on whether The part of the fruit that we consume, the edible part of the fruit, does it form from the ovary or does it form from some other part of the flower? For as long as the edible part of the fruit is forming from the ovary, we call it a true fruit. If it forms from any part other than the ovary, like the thalamus, you call it a pseudocarp or a false fruit. Example for true fruits include mango, guava, orange and all of that. Apple, cashew and strawberry are examples for false fruits. The part of the fruit, the fleshy part of the fruit that we consume does not come from the ovary, it does not form from the ovary. Instead, it is formed from the thalamus. If you look here, the core of the apple that we throw away, this is the actual ovary. The part that we consume is the thalamus. Even in strawberries, the fleshy part is formed from the thalamus. These tiny, somewhat grainy structures that you find on its surface, that is its actual fruit and that is called the achene. Okay, so this is the actual fruit of strawberries. Next, we have another type of fruit called parthenocarpic fruits. Normally what happens after fertilization, ovules will transform into the seed and ovary wall will transform into the fruit. Correct? Over here, fertilization itself does not happen. Because fertilization does not happen, seed formation does not happen. But even in the absence of fertilization, fruit formation will happen. Since fruits are forming without fertilization, you don't find seeds in that fruit. So such fruits will be seedless. In nature, you can see banana does not naturally come with any fruits, right? Any seeds. So that is naturally seedless. But you can see some varieties of grapes that are seedless. They are induced to be parthenocarpic by spraying some plant growth regulators or plant hormones on it. They are made to form seedless. I recently saw about or read about one variety of watermelon also that is seedless. Okay, so more pulp, lesser trouble, right? So it is preferred. Yeah, so this can be induced through growth hormones or it can be natural also, as in the case of bananas. We're almost reaching the end of the chapter. Now we'll move on and talk about apomixis. There are last two concepts left in this chapter. Apomixis is like super interesting. You know, humans mimic other humans, right? You've all seen Sunil Grover, I suppose. He imitates Amir Khan, Shah Rukh Khan, every person. He can just transform into that person and he mimics them. He, pretends to be them. Similarly, you can find mimicry even happening in plants. So this form of mimicry is called apomixis. It's a wonderful phenomenon, very unusual. So here what happens is asexual reproduction. This is the mimicry artist. This is pretending to be sexual reproduction. Which can be any actor, like it could be a Dr. Rajkumar, it could be a Rajnikant, or it could be anyone. Like you've seen a lot of people try and imitate Rajnikant. Every time somebody tries to imitate Rajnikant, superstar Rajnikant, what do? They try and imitate some of his mannerisms, the way he talks, the way he walks, his style of putting on his glasses with that sound, zuk 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 sound, and the way he probably, what is that? The chewing gum that he puts inside his mouth or whatever that is. Whenever a person is trying to imitate or mimic another person, they look at their body language, they look at their mannerisms, they look at how they speak, and they try to do the same thing. And then they will start appearing like that other person. So every time a mimicry is happening, they pick on certain mannerisms and features of the person that they're trying to imitate and they will try to do that. Over here, asexual reproduction is trying to act like sexual reproduction. So asexual reproduction has to pick some features of sexual reproduction and try to do that, right? So the feature that asexual reproduction picks from sexual reproduction is the production of seeds. Normally during asexual reproduction, seed formation does not happen. But here, asexual reproduction without fertilization, seeds will form. See, when I told you earlier about seeds, I told you that in most cases it forms after fertilization. It's a product of sexual reproduction. But here is an exception. Your seed is forming, but sexual reproduction hasn't happened. Fertilization hasn't happened, right? So this is a type of, apomixis is a type of asexual reproduction that mimics sexual reproduction. It is a phenomenon where seeds are forming without fertilization taking place. This can be seen in the members of Asteraceae and grasses. Asteraceae members include sunflower, marigold, and grasses include your wheat, rice, maize, ragi, all of those. So apomixis is the process by which, or a phenomenon by which, seeds are produced without fertilization. If fruit is produced without fertilization, we call it parthenocarpy. Seed formed without fertilization is apomixis. Okay, so this is a type of asexual reproduction that mimics sexual reproduction. There are various ways in which apomictic seeds are formed. One example we'll see. Normally what happens when inside the ovule there is a megaspore mother cell that will differentiate, that will undergo meiosis, give rise to 4 megaspores, 3 degenerate, 1 becomes functional. That gives rise to the haploid embryo sac. After fertilization, the ovule becomes the seed. But in apomixis what happens is this meiotic division itself will not happen. The diploid megaspore mother cell itself will act like the megaspore. The diploid megaspore itself will give rise to the diploid embryo sac. And the diploid egg in the embryo sac will start acting like the embryo. Even without fertilization, the seed formation will happen. Okay, so this is important. This can be of use to farmers. I'll tell you why. I told you about artificial hybridization, right? When you have two different plant varieties, each one having one trait that you desire, you can combine both of those traits into a common hybrid. Now, if you allow that hybrid plant, you get to allow undergo sexual reproduction. Again, there will be crossing over during meiosis. Again, those characters might segregate, right? In the offspring, you may not get that same combination again if it undergoes sexual reproduction. So farmers, if they have to grow hybrid crops, they have to keep buying seeds year after year, because one season they've grown the crops, they obtained the seeds from that plant. If they sow them, they may or may not get the same combination. So if apomictic seeds can be produced from these hybrid crops, it will reduce the cost of buying seeds year after year for farmers. It will make it, more cheaper for farmers. So they're trying to find out which genes are causing apomixis and introduce those apomictic genes into hybrid plants to make sure that they also produce apomictic seeds. If it is forming by apomixis, meiosis is not happening, crossing over is not happening, whichever combination of traits you've obtained in the hybrid, that will be retained in the successive generations too. Okay, so that is one advantage apomixis can have for farmers. Now moving on to the last concept of this chapter, which is polyembryony. It's self-explanatory. Poly means many. Poly means many. Embryony obviously refers to embryos. Normally within one seed you have one embryo. I told you earlier that embryo is the future plant. It's like a baby plant that is sitting inside the seed. Once the seed is sown, the embryo will germinate, and the seed will germinate and the plant will form, right? So normally when you sow one seed, you get one plant sprouting out of it. But in some cases, like in seeds of mangoes and citrus plants like oranges, you will notice that if you sow one seed, you will get more than one plant arising from it. That just indicates that the seed had more than one embryo. Embryo itself is going to give rise to the future plant, right? So this case where one seed has more than one embryo is called polyembryony. There are many ways in which many embryos can develop inside a seed. I'll tell you some ways. One normal embryo formed from the zygote will be there, that is your zygotic embryo. Apart from that, some diploid cells from the nucellus or the integument can penetrate inside the embryo sac and start acting like embryos themselves. So in such cases, An embryo can form from the integument, an embryo can form from the nucellus, and the seed will end up having many embryos. So that is called polyembryony. With that, we've reached the end of the chapter. At the beginning of this unit, there was some information given about Panchanan Maheshwari. So I want to talk about that too, because in the NEET exam, you can expect questions asked about scientists too. So Panchanan Maheshwari was our Indian botanist. He is known worldwide as a very renowned scientist or botanist. He was born in Rajasthan in the year 1904, Jaipur, and he moved to Allahabad later for higher education. He obtained his DSc degree there, Doctorate in Science or Doctor of Science. That was a degree that used to exist back then. And he was very interested in the field of botany, specifically in morphology. And back then, while he was studying, he met a professor. He was an American missionary. His name is Professor Dudgeon, W. Dudgeon. So he was very inspired by his teacher, and then he went ahead and asked his teacher, if there's anything I can do for you, what can I do? So the teacher said, if you want to do anything, then become better than I am. So if you do better than me, then that is like the greatest gift for me. So he gets inspired by that, and he continues to do his research and stuff in the field of plant morphology and embryology. So yeah, he worked on embryological aspects. If you remember, I told you in the beginning when I was talking about pollen-pistil interaction that if a different species pollen grain lands on the stigma will reject it, not allow pollen tube to form, right? So he wanted to find out how you can create interspecific hybrids, like two different species, a hybrid of that. If you rely on pollen-pistil interaction, it will be a failure because the pistil will outrightly reject it. So in order to make sure that the pollen-pistil interaction, to get an interspecific hybrid, he would directly poke a hole in the embryo, 1 or 2 holes, and then introduce the pollen grain through that hole directly into the embryo, directly into the ovary, and not through the pollen pistil route. So he experimented with all of that, and he also insisted that during classification or taxonomy of angiosperms, embryological characters also can be used. So he was very well known for artificial culture of immature embryos, test tube fertilization, this is what I spoke about, intra-ovarian pollination. All of this gave him a lot of fame across the world, and he was also awarded with many fellowships. For example, Royal Society of London, Indian National Science Academy, and many more. And in fact, in the formation of our NCERT textbooks in the year 1964, I suppose, the very first textbooks of NCERT, he contributed in, finalizing the syllabus for biology, what should get into the books, what syllabus can be avoided, and all of that. He contributed in those discussions as well. So that is about our wonderful Panchanan Maheshwari. So that's about the chapter Sexual Reproduction in Flowering Plants. We've reached the end. I don't know since how many hours I've been speaking to walls here. If I do not get like 100k views within 1 hour after posting this video, I will be so mad. So go ahead and share this with everybody you know. And if you have any doubts, you can drop them in the comments. I will answer them definitely. And I hope this helps. If you have any other suggestions, if you want me to practice questions, let me know in the comments. And if you've reached till this point and finished the complete lecture, type done in the comments. That is it. I'm done for the day. I will see you again with another lecture. Please take care. As soon as you watch the lecture, go read NCERT so that all of the information gets retained in your brain for a very long time. That's all for today. Thank you very much. Tata, bye-bye. Take care. All the best.